exposure pathways for most of the climate-sensitive conditions that affect human beings. So as you can see here, we have the first category on the right here where we are talking about extreme weather events and heat stress. These are mostly what I would call the direct impacts of heat and climate change. And then we do have the second category, the exposure pathway, which now involves the indirect mechanisms, the environmentally mediated ones. So we could have conditions associated with air quality, poor water quality, food safety, and then lastly, vector ecology. I think for this region of the world, vector ecology or conditions associated with vector ecology are quite prevalent. Malaria, dengue, chikungunya, and many of the other viruses there which have been characterized within this region. And then, of course, there's the last category around the social factors which could be amplifying some of the direct and the indirect effects. I think the one thing which this publication pointed out is that there's significant gaps in data. And I think we'll just look at that in a short while. So when you look at the availability of data, this is a study which was just published last month in September. Again, from some of the people who've been working around this, there's a big push currently to do what we call attribution. They look at what amount of health risks or health outcomes are attributable to climate change, and they try to classify this across the globe. So when you look at those, the size of the circle represents the number of publications which have looked at attribution. Don't worry about the United States, North and South America. I think that circle is small, but we know why. So please focus on Africa, the Middle East, parts of Europe, and others. Now, when you look at Africa, a lot of the attribution studies that have been done are around heat, or heat effects, and also heat-related injuries, morbidity, mortality, cold-related deaths. A lot of these are forming the bulk, and this, for me, is really a mismatch of what we know is happening on the ground, because a lot of our problems are infectious in nature. So when you see that a lot of the attribution studies are coming up with findings around heat-related injury, majority of those in Africa, that tells you that probably the agenda driving that research probably is not originating from the local situation. We have a number of studies, we look at them in Kenya, but at the global landscape, this is how the data looks like. So this really, it points out that a lot of these are focused on high-income countries, and probably the priority is also driven by those high-income countries. Again, if you look at the Lancet, they usually do like a countdown trying to look at climate-related risks globally. And there are a couple of things which the last report from 2024 is highlighting on Africa. And one of those is that there's almost a 6-fold increase in heat-related deaths and morbidity, especially among the working population. In Africa, that is something which no one is talking about, but it's happening. Then there's the vector-borne expansion. I've tried to list some of the conditions there. Malaria, of course, the regions, there have been several publications about the region of malaria expanding, and then also dengue and chikungunya, because the vectors they are newer vectors, but those vectors are also going to areas which were previously thought to be cooler. And then there's also Rift Valley fever, which is, I think, the new one on the block. We've always known this to be a lowland disease where there's flooding, but we're beginning to see lots of new trends around that. Then there's also food security and malnutrition-related issues. When you get lower rainfall or drier seasons, longer drier seasons, then you get yield declines, for example, on most of the staple food. The one thing which this report highlights is there's an apparent data gap, and they feel that this data gap is much more pronounced in Africa, because when they do those attribution studies, when they do literature analysis, there's a lot of gaps. Most of the African countries do not have continuous data sets which are able to provide or link with the meteorological disease data or mobility and socioeconomic data sets. Again, just to drive that point home, there's this other study which was published. I think this is a report published by the Intergovernmental Panel on Climate Change. They usually do annual assessments, and their 6th report is trying to highlight the kind of data gap that we have in Africa. The first map here shows the research funding. Of course, I think East Africa is doing slightly better, but you can see most parts of Africa, there's very little that is going in terms of climate and health research. On the right-hand side there now is the publications and authorship, again, you can see there's a lot of disparity. So what I want you to focus on is not the green and the dark areas, but essentially just the areas which are appearing clear, because those are areas where there are either data gaps or very little to talk about. And the same is shown down here in terms of adaptation approaches. So just in summary, these two reports show that Africa is climate vulnerable, but we don't, that's not the only problem. So there's climate vulnerability, but then there's also the issues about data, which is useful for planning. And then also Lancet looks at that, and the conclusion is that fewer than 1 in 5 African countries are able to quantify their health impacts of climate extremes in real time using the data which is existing. So for me, in terms of the link between the 3 arms of One Health, talking about human health, environment, animal health, there's a missing link, and that missing link is probably, at the moment, could be attributed to lack of data which links those various institutions. Coming closer home, this is a study which was published again this year by some of our colleagues, Jessica Gerard and team. And just to summarize this, there's also a lot of regional disparity. You know, when you look at Kenya, we have all the climates. We have the highlands, we have the lowlands, we have the hot and dry, we have the wet. And if you look at this, there's also a lot of disparity. There's a bit of a biased publication in the Western Kenya and coastal region. A lot of this is, again, attributable to malaria. These are publications which are talking about climate impacts on health. And there's a lot of regional diversity with a heavy emphasis on vector-borne diseases, both in the Western Kenya, where we have malaria zones, and also in the coastal region, but very little happening for the rest of the country. In terms of the top conditions, it's vector-borne, of course, malaria topping there. And then list down here, we have mental health and heat effects, which are not studied, but a majority of these parts of Kenya which do not have many studies, those are the parts of the Kenya which are usually the hottest. So that's just kind of trying to emphasize the challenges that we have with data, disparities and inequities that we have with data availability across most parts of the country. Part of the work that I do, and this is why I think I got invited to talk in this conference, is that I work with a team from KEMRI and CDC. We've been running a surveillance platform that has been going on for 20 years, from 2006. People started it way before I joined, but that platform has 3 geographically representative populations from the ASALs, the arid and semi-arid lands in Isiolo. That's the site which you can see. I think this is not working, but the site which is marked in orange. And then we have another site in an urban informal settlement in Nairobi at the center there, in Kibra. We have another site in Western Kenya in Asembo that have been collecting data from about 88,000 people. This platform is significant in the work that we do because it's essentially a living lab of One Health. We have various stakeholders who are on that platform. We are doing animal health studies, trying to look at animal public health issues. We are looking at human conditions that might also be originating from the animal sector. And the good thing is that in about a year ago, we expanded that platform to add a little bit of climate data so that we are able to do climate, human health, animal health linkages and be able to analyze the data retrospectively. So the evidence that we are collecting there is able to help us predict some shifts or trends that could be attributable to climate given the richness of the data. So just digging a little bit deeper into that, one of the studies we've been doing is on tracking the SARS-CoV-2 across the country. And this started way before COVID. We've been doing this progressively over time. A lot of the time, most of our data actually corresponds to the national or the what you would call the Kenya spikes and troughs that we have in COVID numbers across the country. This is one of the very comprehensive data sets that comes out of that platform. Another example, I think these ones I'm just picking a few, is part of the data sets that we have is on malaria, and we've analyzed this to try and determine the impact of climate variability on malaria incidence. Malaria is one of the conditions which has seen so much massive investments over time, and the thought around this was we wanted to see whether climate, is it climate or the interventions that are working or that are actually responsible for majority of the cases which are being reported. And what this data shows, if you look at this publication which was done by Brian Nyawanda, is that it's the climate variability that is actually driving the numbers, not so much about the interventions. And one of the things that they're recommending is that before interventions are put in place, maybe the government needs to also try time those with the climate variables, so that they're able to time the interventions when they can work best. The other disease that we work on is Rift Valley fever. This is led by my colleague Njenga. And on Rift Valley fever, we started off with the knowledge that Rift Valley fever circulates in most parts of the country. It's endemic. What we didn't know is whether there was any transmission which was happening in between the El Nino rains, because Rift Valley fever is associated with flooding. So you get most of the cases coming every 4 years when we have the long El Nino rains. So we did some modeling work with colleagues, and what came out is that within the East Africa region, looking at the data for East Africa only, over the last about 8 to 10 years, we've seen some significant increase in annual mean temperatures by between 0.1 to 0.3 for every decade. And that is associated with the decreased rainfall for the arid lowlands, but is associated with increased rainfalls in the highlands. Now, what that has meant is that for areas like Muranga, which is in a very, it's high altitude, no one would think about lowland diseases moving to a place like Muranga in Kenya. And what we realized is that we started seeing several isolated cases of Rift Valley fever being reported in such areas. What this data showed us is that the hotter and wetter highlands, which are initially thought not to be like a zone where we should be getting Rift Valley fever, we started getting lots of cases, up to 76% in parts of Uganda, 43% in Kenya, and this just shows that those traditional areas where these diseases are prevalent have been shifting. So this figure here is summarizing the work that we've done on MERS coronavirus. We started that surveillance in Marsabit way back in 2017. When we went in, we didn't know there was MERS coronavirus in Kenya. It's a Middle East disease. It's a coronavirus found in the Middle East. So when we started that, we didn't know we would find anything. But as you can see here, all those red spikes are indicating times when we are isolating the actual virus from the camels. And what we found is that there's a seasonal shedding from camels, which is also associated with some spillover events in people who work there. So that just means that this is something which is ongoing, driven by lots of climatic conditions. So putting this together, and also looking at the landscape in terms of One Health in the region, One Health can be handled when all collaborators are on the picture. And that is because there's lots of interconnected risks across the disease landscape, human, animal, and environmental health. This really demands for a lot of collaboration among mostly departments which are, the way they're designed, they're mostly working separately and having different reporting structures. No single institution is able to do everything that is needed to control the impacts of climate or even the impacts of climate on emerging diseases. And shared ownership would actually strengthen the collaboration and trust for people to work together. So putting this together again, I try to look at some of the the key things which I feel need to be considered as we look towards moving from siloed, fragmented systems into climate-informed One Health systems. And for me, these are actually falling in 4 groups. There's the integrated surveillance systems, which currently we do have good surveillance systems, but a lot of these are sector-specific. So there's very little interaction between them. There's very little that has been done on predictive analytics and early warning, because that's also one of the opportunities for us to be able to utilize, for example, the meteorology data, climate data, and the rest. There's a lot that is happening on the multisectorial governance and coordination through institutions like the ZDU, and then also having programs which engage communities and take in their input. So this is my last slide. Recently, we put together, through a group of collaborators, we put together an application to welcome Trust to look at climate and health, sort of trying to quantify the impacts of climate on health within this region. This is work which will be going on until 2028. The goal is to establish a climate health causal relationship for some of the common syndromes, And we are going to be using the PBIDS platform, the population-based platform that I referred to earlier. The goal there will be to see how well can we integrate climate and health data so that we are able to get some of these deeper insights. And I think that should be the last one. These are some of the things which we already receive from our literature review, which should be able to help Kenya to achieve and sustain some of the goals around One Health. I think that should be all. Yes, One Health matters because the health of people, animals, and the planet is one single story, and only by reading it or telling it together we can write a more resilient future. These are the collaborators that we work with, and I would like to acknowledge some of them who I know are in the audience here. Thank you so much.
Okay, thank you so much, Dr. Gere. Let's put our hearts together again to appreciate him. So my name is Dr. Danson Mwangi. I'm here to invite our next guest, who is Dr. Mark Nanyingi. Dr. Mark Nanyingi is an infectious disease epidemiologist. Welcome, Dr. Nanyingi.
Good afternoon, everyone. My name is Mark. I'm the technical director of the CGP. It's a locally run organization. But what I'm going to present is over 15 years of my own experiences in research, in academia, and in humanitarian work. And when Dr. Kamau from EPR talked to me to speak, because James Hassel wouldn't come because of the shutdown in the US. So I won't really delve much into the definitions, but for many people who do not really understand One Health, that this is not a new concept, but this is a conceptual process whereby there are a lot of drivers that actually lead to outcome of disease. And the environment usually is at the center of all this, but most of the time we really focus actually on the humans and the livestock. For a very long time, through my work all the way for many years until lately at the WHO, I've been a One Health specialist, and the focus has been actually to try and understand what really drives disease. We have in this room, many of us, we are having our computers, we are driving cars. We also have issues of traveling. I think many people have traveled from wherever they have come from using flights. But also we have agriculture, driving the use of fertilizers and so on and so forth. So clearly, it's an intricate relationship of ecosystems that you couldn't really isolate, that one part of it is actually responsible for that. And I think Dr. Ngere had just finalized his slide by actually showing that is a relationship. So I won't really emphasize on that. So some of these graphics, they're of course borrowed, but some of them actually we build upon them with the proprietary ownership of the WHO at that time. I want to come home, because this is what I've been doing for almost more than 15 years, that Kenya has actually transitioned in a lot of sense, that we have a functional National Public Health Institute. And the National Public Health Institute actually is embedded at the force of bringing animal health, environmental health, and the human health in one perspective. The ZDU, what we call the Zoonotic Disease Unit, has been existing since 2012, when a memorandum was signed between the Minister of Public Health at that time and the Director of Veterinary Services. But this has evolved over time, and many sectors actually have come on board, but the central focus actually has been the veterinary workforce. And how we've been trying actually to enable the ZDU to work is that there are various technical working groups where most people actually belong. I mean, somebody like Isaac and myself, I think we're very conversant with this. But also we are bringing in academia, we're bringing in research, And the essence is how does actually this flow down to the subnational level, which we call the county level. Kenya has a devolved health system, and what we've tried to do over the last, so we started what we call the County One Health Unit while I was working for the CDC in 2015. And now we have evolved actually from the initial 5 pilot areas where we started doing the County One Health Units, and now we have 22 County One Health Units in Kenya. The County One Health Units, what they do is that they are the front face of actually responding to outbreaks that have potential spillover, whether it's from the environment or from livestock. So you have veterinarians, we have public health experts, we have people from disaster working together actually to respond to that. And these technical working groups are actually confluence into now what's the driving force in many of the endeavors that go into even resource mobilization in the country. In this journey, which was started way before probably most of us actually joined this, I think we've been very fortunate to be halfway on this journey to participate on the success stories of Kenya in terms of actually building a framework that is reliable in terms of responding to infectious diseases that have potential for One Health outbreak, for One Health. But I'll just skip way ahead to this journey, and I'll say that the 2012 phase, after the huge outbreak of Rift Valley fever about 6 years before that, led to concerted efforts actually to actualize this into a dream, and it became a reality. And now we were able actually to develop the One Health strategic plan, which expired this year, and I think we're working with other groups of people to try and have another document that charts ahead the country's ambition actually to tackle specifically One Health issues and zoonosis. But I think the biggest milestone actually came last year, where we were able actually to develop the Marburg virus disease response plan and a 72-hour response plan. And this was actually embedded in our One Health approach, whereby I think the animal health sector took a center stage, and also just trying to understand the transmission dynamics of how Marburg and most of these viral hemorrhagic fever spread across board. But I think this year, we are going to launch 2 key documents. And in the next week, I think the Rift Valley fever contingence plan that has been taken a long time actually to finalize, I think is going to be developed. I think it has 13 SOPs that address from case management to surveillance, and I think these SOPs are actually grounded on One Health. But I think just last week, and actually in the next few weeks, we are going to roll out what we call the decision-making tool. So the decision-making tool for public health emergencies is also anchored on a One Health approach, and I think it brings about the stepwise way of how to respond, what to do, and who is responsible for doing what, so that we save on time and resources on how to respond to actually diseases that jump across species. One of the key stories that have succeeded is implementing what we call the IDSR. So the IDSR actually works on a framework of actually signal detection, and these actually, maybe if we have another session, we could actually demonstrate this. Is an app where you have to do triaging and filter out most of those signals. And risk assessment is very key. So you have the joint risk assessment, which is a concept of bringing together all these systems. But then finally now you have to take action. If it's rabies, how are you going to make action to put the animal vaccinations at the center of this, or Rift Valley fever? And the IDSR is actually an endeavor that is being driven by the WHO AFRO, but it actually helps to list 46 priority diseases in the country, and some of these diseases actually have specific signals. For example, I've just picked out a community event-based surveillance system signal number 5, which picks actually animal signals or zoonosis signals, and is actually in real time be able to transmit to a dashboard, and the national office can actually make decisions based on the evidence that is being provided. So this is a game changer, but it's also improving. And I think when you have the PS speak here, we are very grounded in actually trying to digitize most of these tools so that we have an easier way to make a decision and actually save on time and actually human resource. But what we've been doing actually, we have as a country led in many of the One Health efforts. And the Pandemic Fund, which actually was an idea from the World Bank with the WHO as the governing body and the technical advisory panel, has been floating for 3 rounds. We haven't succeeded as a country for the first 2 rounds in 2023 and 2024. And I was fortunate enough to lead all these applications, including this year's application, which was the 3rd round of application of the Pandemic Fund. And the Pandemic Fund is squarely embedded in a One Health approach, whereby we are looking at strengthening the lab capacities, but also improving on how quickly can we optimize our early warning systems while we build capacities of frontline healthcare workers. So we put in, for this year's grant, $25 million, and we are crossing our fingers that probably next month we are going to celebrate to have finally actually won this for the country. But also we have participated to support the African region in actually seeking for a combined value of actually almost $200 million from the various entities. So we might get some, we might not get some, but I think our effort is that if we get some of this funding, we'll be able actually to do a lot of work, not just in Kenya, but actually in the region to strengthen these three areas. Rift Valley fever has been talked about here, and Rift Valley fever is one disease that I walked along for about 10 years, from my master's to my PhD to my postdoc. And last year, actually, while at the WHO, we had an opportunity to respond to Rift Valley fever outbreak in Wajir and in Marsabit. But what you notice is that when the human cases are seen, obviously the animal cases already have happened, but probably they were not just detected because of the subclinical circulation of the virus. So what the One Health approach actually helps is that you have actually the human sampling that is being done actually for the PCR and RT-PCR to detect actually the IgGs. This is actually like the infection, recent infections, but also animals are sampled. Sometimes actually you will be lucky to detect the IgGs or the IgMs. In all this is embedded at the community level, whereby, because these are acute febrile illnesses, there's an opportunity actually also to differentiate this diagnosis with malaria. And so this was a very good success story, and the paper is almost out. But one of the things that I'm very proud actually to be associated with, and I've also seen Professor Bukachi here, is like a couple of years ago, I think it was about 2016, around there, in Baringo, I think we were the first group of people actually to churn out a policy brief on Rift Valley fever, and this has guided the country going forward. So we had 2 policy briefs on malaria and Rift Valley fever, and I think they continue to be improved on by the country and actually inform many of these areas. So quickly, I'll just share the efforts at the transboundary. So last year, with support from the UKHSA, actually we were able to do a cross-border simulation exercise. This is a tabletop simulation exercise, and it helps actually the 3 countries to address some of these issues. And then back here in Nairobi, we enabled to sign an MOU between IGAD, WHO, and the African governments actually on how to move forward and enhance surveillance mechanisms. And then lastly, we have built actually mechanisms of cross-border collaborations. So first of all, to strengthen the cross-border collaboration, improve diagnosis, work on the community level, align emergency preparedness plans, and then finally actually sustain collaboration mechanisms across these countries. So this is a big initiative that is country-driven, but also is led and supported by IGAD and many of these other entities, including the WHO. And then finally, as part of the work for my postdoc, actually we were able to scope a very big review of what happens in the Horn of Africa. And it was quite interesting just to see people churning out very many publications. Quite a ton of publications, but the question has always been, why are you publishing all these papers? You want to be a professor like most of you want to do all these things. So we want actually to translate research into action. And I think that is the One Health action that we want to see, that after seeing all these tons of papers across these countries that actually cut across those domains, and you can see clearly the animal health sector dominates, what next are we going to do? So some of the challenges in implementing most of these things is, Isaac has already talked about them, the fragmentation and silo mechanisms, the resources are not available, lack of workforce, the surveillance systems are torn apart, and the community are not very enabled. Our next frontier, and what we are churning actually into, is actually building epidemic systems that are powered by AI. And what we are looking at actually from this predictive modeling that we've been doing for very many years, how can we simulate diseases in real time and actually be able to make these decisions without actually walking or putting the boots on the ground. And I talked about this tool. So this tool is based on what we call the IHR 2005 Annex 2 of the WHO. It helps you to make a decision how to escalate your response and not spend more resources. So you can decide actually to contain the outbreak within the county or probably seek for the national support from the NPHI. So it's a snapshot of it, but we have a user guide manual that actually is going to be launched, I think, in the next few weeks. My call to action as I conclude this is that we need to integrate by actually continuously trying to work across disciplines. we need actually to strengthen the capacity building. For the young people, they're actually at the crossroads of career. I think One Health is one of the most amazing areas that you can actually, think about, because you'll talk about mosquito trapping, but then you'll go back to the lab, and then you'll go back actually to do sampling. Communities have to be at the center of this, and we've seen actually how Kenya has been at the forefront of this. And then finally, right now we are having a very big outbreak of cholera in some parts of the country. And in this picture here, 2 years ago when we were responding to the cholera outbreak, this picture has all the disciplines that we are talking about. It has a disaster guy, it has an animal health expert, it has a public health officer, it has a medical officer, and all that. So for me, my clarion call is that One Health is the future for us to tackle emerging and re-emerging infectious diseases. Thank you very much.
Thank you so much, Dr. Mark Nanyingi. He was actually one class ahead of me in vet school, so a good time to also reconnect. So our next speaker will be Professor Geoffrey Sewell, who is online. And Geoffrey is from the University of Michigan in the US, and he'll be talking about how AI can be used to strengthen pandemic preparedness and response. So welcome, Geoffrey.
Yeah, thank you for the kind introduction, and it's my pleasure to join you all. I'm originally from Kenya, and I would have loved to be there in person, but as you know, travel can be quite hectic sometimes. And so today I'll speak about some of the work we're doing around developing generative AI approaches for antiviral drug tests for during clinical trials and was safe in humans, but failed in actually treating the disease that was the target. And yet that same molecule might actually be beneficial for another virus that was not considered in that particular trial. So this could be a way to, easily fast track the development of drugs for a new virus. The other thing, of course, is that with the viral proteins, you could begin to do antibody design. And we've seen tremendous advancements in how AI can generate antibodies from scratch. Conventionally, discovery of antibodies, requires use of animals and animal models to be able to screen, or even like taking convalescent sera to try and identify protective antibodies. But with AI, we could begin to design these antibodies just knowing the protein structure, which also can be generated by AI. We could use that protein structure to design an antibody against that protein target. And also this could, of course, inform vaccine design, which AI could help in identifying both B cell and T cell contribution to think about how to perform drug development in a way that considers human viruses as well as those in animals and plants, and see whether this could lead to a more effective way of controlling infectious diseases. And with that, I thank you all for this opportunity. And thank you to the organizers.
So thank you so much, Dr. Siwo, for your presentation. Our next
Yes, thank you.
presenter-
Thank you all.
Okay, thank you and welcome. Our next presenter is Professor Salome Bukachi. Professor Bukachi is from the Institute of Anthropology, Gender and African Studies of the University of Nairobi, and she will present on gender. She will use a gender lens to understand women vulnerability on zoonotic diseases context. I must say, Professor Bukachi is one of the persons who inspired me to pursue medical anthropology as a career. Welcome, Professor.
Let me get this right first. So good afternoon, everyone. It's a pleasure to be here, to be able to just interact with researchers from different disciplines to discuss aspects of health. He's already introduced me, so I'll just skip that. So when we look at the aspects of One Health, Dr. Nanyingi had already brought in the concept of One Health, and looking at it from the aspect of that integrated perspective of looking at health, taking into consideration the environment, the human, the animals. But we also look at it in terms of the cogwheels that drive the aspect of One Health, looking at communication, collaboration, coordination, and capacity building. And all those work together towards producing a healthy nation, a healthy planet, healthy animals. But one of the things that we have that's cross-cutting is the gender, equity, and social inclusion, besides also the social and behavioral aspects that are quite critical in terms of managing disease. And right now, when we look at our theme, which is about innovative research, we need to ensure that our innovative research is also equitable. And so I will just take us through a bit of what gender is and how it can relate to aspects of disease control, disease management. The key component in terms of how we do our work and how we do our research is about moving from gender blind to transformative research. So a key question we ask ourselves is how can we harness our knowledge, understanding, the experiences of people, pathogens, health innovations, and technologies to improve that intersection of human, animal, and environmental well-being while bringing about socioeconomic and cultural transformation? And how do we move health interventions from being gender blind to being accommodative and transformative? How do we ensure that the innovations that we are coming up with, the research we are conducting, is actually equitable towards the communities that we serve? And as we look at it, what has gender got to do with One Health? So where we are is we want to move, when we look at the pictures down there, we see what we have right now is two people wanting to watch a football match. And each of them, one has more than the other, while the other one has less. So of course, the one who has more, the taller one, is able to see better than the person in the middle and the lower one. And that's what we call the reality. But it's about moving from that reality to moving to equality. And equality means giving everyone an equal chance to participate. And if we give everyone an equal chance to participate, from the second picture, we realize that some people, even if they're given an equal chance, they may still not be able to participate or see the football match because of the differences that exist in them. And so we are talking about equity, where everyone gets the support they need that can help them see the match. So that means that the person who is at the lower level gets enough support to enable them watch the football match, which is a good place to be when we talk about equity. But eventually, we want to remove the barriers that hinder everyone from seeing the football match so that those barriers are broken down. And that's the aspect of the gender transformation. So why does gender matter when we are talking about One Health? As we've looked at it, there's that interconnection of the human, animal, and the environment. So gender shapes how we experience disease. It shapes exposure when we are talking about vulnerability and risk. And it also shapes access to care, because not everyone has an equal access to health care. And so we recognize that the different gender roles could increase vulnerability because of the various aspects of where people are engaged in, whether in agricultural activities, giving care, or working within the informal markets. They increase vulnerability of the community. So we'd want to move towards a gender agenda where we embed gender to enhance prevention, disease prevention, preparedness, and response. And we also take cognizance of the fact that addressing gender issues is one of the sustainable development goals. And so if we are able to push our research towards moving to help achieve the sustainable development goals, then our research is already speaking into the policies that exist. So gender analysis, in the work we do gender analysis to help us unearth the inequalities that exist in our interventions and how we relate to communities. So I've worked a lot in the trypanosomiasis space, looking at the social, the cultural aspects of trypanosomiasis. And so when we look at the gendered pathway of trypanosomiasis, we see the aspects of the vector-borne diseases. It's a vector-borne disease, and we find that men's exposure is mainly through their roles in herding, hunting, or working within the SESE-infested areas, while for women, the exposure comes in through the caregiving they do, either taking care of the livestock or food preparations or taking care of other sick people. And all these have different implications to the vulnerability or the risk exposure to trypanosomiasis. And of course, we can also apply that to other vector-borne diseases like brucellosis and Rift Valley fever. So we note that also men and women experience disease differently based on the different aspects. So for example, household and social responsibilities may hinder women from going to hospitals in the early stages of the diseases. We know that, and we have seen this when we look at trypanosomiasis, where women were not going for checkup quick enough because of their social responsibilities in the home. While for men, we also realized that there were also delays for them to go for diagnosis or even for checkup, and this was related to the gender norms in terms of the level of endurance, that there's the tendency towards men to want to be strong and to keep being strong, and by the time a disease, they're going to the health facility, the disease has gone to quite a late stage. So that also has implications on how they present at the health facility. Then for women, one of the things that came out when we were looking at trypanosomiasis was lack of resources that sometimes may hinder their prompt access to health treatment or even care. And they have to depend a lot on the men in their households to be able to provide the finances. And that also becomes something that we need to take into consideration when we are looking at how the experiences of disease from a gender perspective. And when we come to control, because when we look at trypanosomiasis, there are aspects of also control that are involved. The key questions we have asked ourselves is who decides what control measures need to be implemented in a household, and who participates in those control activities. We realized that training many times excluded women, and this is because when intervention, people who are carrying out interventions go to the community, many times they're working with the household head or the main farmer in the home, and so you'll find it's the men who turn up for the trainings. Yet we know that women are also equally involved in the aspects of livestock care or aspects that would relate to disease control. We also realize that sometimes women's knowledge is undervalued, especially in communities where we come from, where the patriarchal system elevates the man's knowledge more than the women's role. And in some communities, women are meant to be at the back and not at the front. So those again become aspects when we are looking at gender in disease control. So when we look at gendered control in relation to vector control, we realize that when it comes to decision-making, participation, and training, it's men who have more access and their knowledge is more valued as opposed to women who have least access to these aspects of decision-making and also limited in terms of the knowledge, because we realize that the trainings that happen, they are not always part of it unless one is very deliberate in that. For trypanosomiasis, some of the vector control measures that are there is about targets, livestock baits, pour-on, and treatment of livestock. And again, this requires funding to be able to access these elements. And again, the gendered variations come in that. We've talked about decision-making on whether to control, how to access the equipments that are used for controlling, and the responsibility of animal health care. We've talked about information dissemination, where we see that it's mostly men attending meetings, but also we find that many times we are holding meetings when most women are taking care of the household activities. And so these are things that we need to be conscious about when we're dealing with communities to ensure that there's equity in terms of our interventions. So to enable us embed gender within the One Health, we need to use intersectional participatory approaches that looks at different gender, for example, looking at women not just as a homogeneous group, but there are different categories of women: adult women, young men, young children, among that, so that then our data is able to address the different categories of groups in those gender groups. We need to co-create with communities and especially women, so that then our interventions are relevant, and then collection of gender disaggregated data. Many times we bulk our data, and so when it comes to developing interventions, it's a one wholesome thing that may not be relevant to all different gender groups. And then, of course, aligning with the aspect of justice to ensure that structural barriers are removed and sustainability. So even as we conclude, gender is not just a component, but it's a central, it's not peripheral, but it's a central and a core aspect to effective One Health approach. And so when we address gender inequalities and women's vulnerabilities, these help to improve on the health outcomes for all. So it's about moving from inclusion to transformative aspects to enable us achieve One Health by centering gender equity in One Health. Thank you.
Thank you so much, Professor, for that wonderful presentation. Many years ago, I used to think gender is about men and women, but I came to understand that it has more to do with our gender roles. I mean, what men do, what women do, and how this exposes people to different experiences and exposure to diseases. Our next presenter, I am advised that she is not online, but her presentation is recorded. So let's have the presentation from Professor Stephanie. Stephanie is a molecular ecologist working on zoonotic diseases and animal pathogens from the University of Washington State University. So let's have the recorded presentation.
Input. So today I'll be speaking on predicting viral emergence through ecological theory, evolutionary insight, and data synthesis. And in my of Chiroptera with these coronavirus, and as we go in, how many shared viruses in yellow school? Hello, my name is Dr. Steph Seifert, and I am an assistant professor at the Paul Giello School for Global Health University in the U.S. I'm in person with you today, but I'm very grateful for the organizers for allowing me to record this this presentation. And I hope that you will reach out if you have any questions or comments or thoughts. I'm very open to collaboration and appreciate input. So today I'll be speaking on predicting viral emergence through ecological theory, evolutionary insight, and data synthesis. And in my lab, we study viral zoonoses primarily, or viruses that are spread between humans and animals. And the reason that we focus on viral zoonoses is that roughly 75% of emerging infectious diseases are zoonotic in origin. And so in my lab, we study cross-species transmission of viruses, which we believe is a fundamentally cross-scale problem. And at the ecosystem level, we're interested in host behavior, biogeography, and population dynamics that shape the likelihood of exposure to novel viruses. Yet the outcome of those types of exposures, or whether a virus can infect, replicate, and transmit, depends on molecular interactions. And this includes things like receptor binding, immune evasion, and compatibility with host cellular machinery. To bridge across these scales, we integrate tools from ecology and evolutionary biology. And we hope that this multi-scale framework will allow us to better predict and ultimately mitigate the emergence of zoonotic threats. And so I want to focus for just a moment on the host virus network of coronaviruses. And so this is my work my PhD student Irene Karegi has been putting together. And when we look at coronaviruses, specifically around the edges here, we have our hosts, which are the nodes, these little dots. We have them in blue if they're a wild species, or orange if they're domestic. And then the lines that are connecting these dots are actually shared viruses, individual shared viruses. And so the thickness of the line corresponds directly to how many shared viruses there are in this case. And what we see with coronaviruses is that Chiroptera or bats, we have a lot of shared viruses within this taxonomic group. So you can see how thick these lines are, and that's just shared coronaviruses between different host species within bats as an order. And when we do have sharing outside of Chiroptera with these coronaviruses, we have some that flow to humans, and quite a few that are actually flowing to domestic livestock. So these orange nodes over here, these include cattle, camels, alpaca, a wide variety of artiodactyls that are domesticated, swine, for example. And so we see that there's a lot of sharing specifically with domestic livestock that are artiodactyls, and also sharing directly with humans. And then we also have sharing of coronaviruses from our domestic artiodactyls to humans down here in the bottom. We also share quite a few coronaviruses within our taxonomic group of primates. We share a lot of coronaviruses with other non-human primates. And so this is starting to give us a peek into the idea that ecology, or how we interact with animals, can lead to more sharing of viruses. And if we go a step further from that host virus sharing network, and we really dive into the specifics of a virus, we can actually pull a lot of data from existing literature. And so for the last 2.5 years, I've been working with Dr. David Simons to extract all surveillance data for arenaviruses and hantaviruses from rodents. And this has been very fruitful. We have been able to pull data from the past nearly 50 years. And we have over records from over 570,000 individual animals that have been sampled for these two virus groups. And we have data on almost 700,000 individual molecular assays, including ELISA, isolation, and PCR. And we've connected these as well when available to sequencing data. And so this is an incredibly valuable data set for us now to ask questions and test hypotheses that are well beyond the scope of those initial papers, which are much smaller studies in time and space. And when we plot all of our surveillance records, we also kind of immediately start to identify some gaps in our surveillance. So in North America and Canada, we have almost no surveillance for these two virus groups, even though we know that Cynombre virus does cause infections in humans in Canada. We also see that there's not much surveillance for hantaviruses in Africa. The focus has primarily been on arenaviruses, largely because of Lassa. And as we go into Europe, we see much less surveillance for arenaviruses in general. So I think that we're probably missing a lot of viral diversity for these two groups, just due to where we're prioritizing those surveillance efforts. We can also start to see that this old paradigm that hantaviruses have a really close evolutionary history where it's one host, one virus. It doesn't appear to be the case as we start to explore the prevalence for hantaviruses across different taxonomic groups of rodents. For instance, this is Andes virus. Andes virus causes severe disease, hantavirus cardiopulmonary syndrome in South America, and is, I think, a respiratory virus that we should be following very closely. We see that there's actually high prevalence in several taxa, and they're phylogenetically distinct, that these are not only, one species and one virus. This virus is actually found across the rodent tree in South America. And so this is also helping us to identify priority taxa that we haven't sampled yet, and give us an idea of the true breadth, host breadth for some of these viruses that are important as viral zoonoses. And when we start to think about how we can take those ecological data and make predictions about compatibility, we know that we also need to understand something about molecular compatibility. And so if we can pull data from sources like this is an experimental data set where they are testing receptor affinity between the coronavirus spike glycoprotein for SARS-CoV-2 variants of concern, including Delta and Omicron and the prototypical strain, and their compatibility with ACE2, the receptor, from these different host species. And we can see that some species are fully incompatible across the board. So we have very little compatibility with the little brown bat for Omicron, for example. Guinea pigs appear to be incompatible with this virus, whereas cats are very compatible. Mink, especially with Omicron, very compatible. And so this information can also be leveraged to help us build more informative models on host virus compatibility. And so I want to focus a little bit on some work that we published recently in communications biology. This work was led by Katie Sang, who is a graduating next month PhD student at WSU. And so we were interested in identifying reservoirs of orthopox viruses. And so orthopox viruses, we have a very famous member of this group of viruses, which is Mpox. And we have the emergence in recent years of two independent lineages where we have higher human-to-human transmission of Mpox, and this is a pandemic threat. But we have a pretty good idea of the reservoir for the clade 2 Mpox now, likely rope squirrels. It's possible that rope squirrels are also the primary reservoir for clade 1, but we don't really know. And as this virus is spreading globally, we also don't know which other species around the world could become reservoirs for Mpox internationally. In the Americas, we also have the emergence in recent years of Alaska pox or boreal pox. And this virus, we have 2 case fatalities associated with this virus, and absolutely no idea where this virus comes from. And orthopox viruses have variation in their host breadth. So acromelia virus is also known as mousepox. This virus is primarily associated with Mus musculus, the common house mouse, and not really any other taxa, whereas Mpox has been discovered in all kinds of different rodent species and non-human primates and humans, and in 2022 also specifically an Italian greyhound. And so we started out kind of replicating previous work done by Barbara Hahn and Jen Becker and others on developing a host trait model to predict orthopox virus reservoirs. And so this is employing a machine learning algorithm called boosted regression trees. And then we collect data from different sources. So for host virus association data for orthopox viruses and their hosts, we pulled the information from the Virion database, which is the largest host virus association database available. And then we pulled host trait data from the combined database. And so this is largely life history traits, but includes other extrinsic traits as well for host species. And so the idea with this is that we treat orthopox viruses as kind of a monolith where all of their traits are held fixed. But what varies are the host traits. So life history traits, for example, associated with the host species. And so the database actually looks like this, our data table looks like this. And so we want to know the probability that any fixed virus can interact with any specific host species. And so in thinking about this, we were concerned that we're missing all of the variation that's present for orthopox viruses, right? We know that Ectromeliopox does not have the same host breadth as Mpox, and we actually want to know what are the potential reservoirs for Mpox, or what are the potential reservoirs for boreal pox specifically. And we want to train our model based on viruses that we do know something about, and then try to project that into viruses that we maybe don't have as much surveillance data for. And so we decided to incorporate our virus traits into what we call the link prediction model, where we're predicting a specific virus with a specific host species. And there's an interesting kind of thing about orthopoxvirus evolution. We call it the genomic accordion because orthopoxviruses are a very large genome compared to coronaviruses. We have about 200 viral genes, over 200,000 base pairs. And these viruses employ, as an evolutionary strategy, these insertion deletion mutations for accessory genes that antagonize the host immune system. And so we created this matrix of gene presence or absence across all of the viral genomes that we could link to a specific host species. And so this included experimental infections, field surveillance, and also Mpox genomes from outbreaks in humans. And so we created this matrix, and then we did a principal component analysis for dimensionality reduction. And we included those principal components, which we now call our viral traits, into our model. And when we compare our link prediction model, we see that it outperforms our host trait model, which is based on host traits alone. And so on the right here, these two are just two kind of versions of our host trait model, one based on PCR positivity, the other based on virus isolation. And then our link prediction model, either including or excluding vaccinia virus, but in both cases, we find that the model performs very well. And so then we wanted to look at the traits that have a large relative influence on our model performance. And with our host trait and isolation model, we see that citations are very important. We cannot build these models without data. And also that dispersal and gestation lengths were highly ranked. But when we look at our link prediction model, again, we find citations are highly ranked. We need data, regardless of which model type we have here. But we also find that our top, below citations, our top 4 important features were actually our principal components of our viral traits. And so we wanted to dig into this a little bit more. And what we find is that the principal components that were highly ranked include a lot of genes that are involved in host interaction and immune evasion, virus replication and gene expression. We also have some that have an unknown function. And so these genes that have no known function for orthopoxviruses are coming up as highly ranked and having an importance in determining whether that host species is compatible with that specific virus. And so this is a list now of genes that we can provide to our colleagues who do more bench work and ask them to try to start prioritizing, figuring out the function for some of these viruses, or trying to understand mechanistically how specific genes in these principal component sets influence the outcome of exposure to different viruses in novel host species. We were then able to develop a map. And so with our map, we've shown now our predicted undiscovered hosts and those distributions across the world. And what we find is that there's a lot of host diversity for predicted orthopoxvirus reservoirs around the equator, which is perhaps unsurprising. But what we were also interested in is seeing how this map of kind of predicted undiscovered hosts overlays with vulnerability. And so the Mpox vaccine that we're using today is actually the smallpox. It's the same as the smallpox vaccine. And that's because there's cross-reactivity in the vaccine for orthopoxviruses. And so if you're protected against smallpox, you have some efficacy and protection against Mpox as well, and other potentially novel orthopoxviruses. And what we see is that the vaccination coverage in the areas where we have a high diversity of undiscovered orthopoxvirus reservoirs, it overlaps pretty well. And in a sense, I think this model is telling us not only where we should look for undiscovered hosts of orthopox reservoirs, but it's also telling us where vulnerable populations are to emergence of novel orthopox viruses that we could do a better job of protecting. And I want to take a little detour to another virus group that is kind of surprising in that we don't know where this virus comes from. And so we're almost 50 years out from the characterization, the first isolation of orthoebolavirus aierense, also known as just Ebola virus. And when we look at the CDC figure, we can see that it's worked out, right? That we have the virus circulating in bats, it spills over into duikers or non-human primates, and then it's indirectly transmitted to humans. But this isn't that well worked out at all. We actually have never isolated Ebola virus from a wild bat. We do have a lot of serological data suggesting circulation of ortho-Ebola viruses in wild bats. And so with that, we want to figure out when we should be sampling in time to actually confirm a reservoir for orthoebolaviruses. And so we employed an SIR modeling framework, which I think most of us are more familiar with than we'd like to be after the COVID pandemic. And I worked with Aaron Clancy and Scott Newsmer to adapt this model so that we could predict in time when the peak infectious virus is in a wildlife population based on just serology data alone. And so the first step in this is for us to define our different parameters. We include things like rate of recovery, rate at which the antibodies decay, et cetera, birth pulses, which in many bat populations is seasonally fluctuating. And then we solved for our infected class to get at infectious peak in a population over time. And so we applied this model to a data set on serology from Eidolon halvin bats, showing circulation of an ortho-Ebola virus in this population that has an annual birth pulse in time. And with our model, we're able to show that about 2 months following the peak in the birth pulse, we actually have our peak in infectious shedding. So in time, if we want to actually go and get a PCR positive for this virus, we think the time to do it is about 2 months following the birth pulse. And so we think this model is really helpful not only for prioritizing when we sample in time, but also for predicting high zoonotic hazard, or when prevalence of infectious virus is very high in a wildlife population. And so with that, I just want to summarize that using different modeling approaches, using data across disciplines is I think, really integral in improving our ability to forecast or predict emergence of viral zoonoses. I want to thank both my funders and collaborators, including folks at the Verena Consortium, the Paul J. Allen School for Global Health, University of Idaho, and the Institute of Primate Research. And if anyone would like to reach out to me, I'm available through email at stephanieseifert@wcu.edu. And with that, thank you for your time.
Dr. Mas, I think I've lost the sound from your end. Fortunately. I maybe drop off and then try to restart again. Dr. Massey?
We can hear you.
Okay, cool. Now I can hear you now. Do you want to do intro again?
Okay, fine, I can do the intro again. So Dr. Joseph Kamau is the head of One Health, as I mentioned, and is the one who put together this session. And we are grateful for you for selecting wonderful speakers. And he'll be talking about how landscape changes drive emerging public health threats. Welcome, Dr.
Thank you, Dr. Massey. Good to hear from you. And I'm sorry, maybe you cannot be able to see me because of the bandwidth I'm using, but bear with me. Maybe I'll try towards the end of the talk. Getting just more to what maybe the previous presenters have just spoken, we live in a very changing landscape, extremely exacerbated by the climate change. So many things are happening, changing how the livestock production, agricultural system are being carried about. And all this kind of goes to do with what the previous presenters talked about, potential pathways, exposure, spillover, and how human, animals, or environment becomes contaminated with the potential pathogens along the way. And so because of this one, we try at the One Health Center to see whether we can be able to develop what we call the disease early warning system. So how possible ways to detect at the community level, and how this one can be escalated to the national level, and how that will be able to be responded timely at the right time, so that we can be able to limit the extent of the damage or the community effect. So to do that, we know the threat that once the drought comes or the floods comes, there's quite a lot of movement of those animals, and when animals do not move, or then they do not kind of migrate, we ourselves migrate. And when sometimes animal migrate, also they migrate with the vectors. And we know vectors are basically one of the biggest pathway of, or the go between animals and humans. So we have seen in cases where an elephant, when it moves from one area to another area, it carries millions of sesaflies with it. And communities have associated that movement or migration of some of these animals do cause or associate or correlate with an occurrence of an event of a disease. And some of that one's to do with the sandflies on leash. We have seen also that one on trypanosomiasis, which is caused by SESE, but also recently you have also seen that one with the mosquito-borne related diseases, including the RVF. So when that happens due to land use changes, when that happens due to environmental changes, or due to the extended drought, then that correlates with what events or occurrence of outbreak of some of disease in the humans that we get reported. And I've just, as the previous speakers have spoken, using the One Health approach, we try to see then where's the best place, either upstream, downstream, or in between. Where could we get maximum intervention that could be able to reduce the scale of the output, or maybe the infection downstream. And so from this picture, the One Health here, we see as Dr. Stephen has mentioned just a few, how the human share, the virus being shared between animals, and the livestock, both livestock, wildlife, and humans do share quite a lot of things. For all these one, it leads to 2.5 cases of infection that are reported globally every year that do infect or do affect humans. But out of that one, 2.7 deaths annually occur that are caused by the zoonosis. And this is just that what we can be able to record. And I remember a previous speaker that did mention that we have gap in terms of databases. So we don't know the number of other cases that I will sit down as we proceed. Some of the data that we miss we may be having more than 2.7 million deaths of humans every year, and maybe there are even 4 or 5 million. But because of lack of the data, how it's recorded, some of the places that it occurs may be in very remote areas, so it's not captured as the right way we would want to capture it. So we have this missing, and so we sometimes we find that the people trying to implement the policies do not see the essence, because some of those areas are missing, and we know that our neighbors or some areas that is missing, to try and actually underscore the importance of the zoonotic disease in human cases. So what was captured last year in 2024, Lancet did report that 1 in 4 global deaths are due to modifiable environmental factors. And when you talk about this one, as mentioned by the first presenter, there are quite a lot. This includes temperature changes, rainfall, and other modifiable environmental factors that do occur. And that one correlates or is associated with the 1 case of death in every 4. So basically, we're talking about 25%. So in Kenya, we have like around 1,300 of deaths, that 1,300 deaths every day. So you could assume maybe 300 of the deaths that occur daily in Kenya are due to modifiable environmental factors. And we know that 80% of human pathogens are environmental modifiable. So using the One Health lens, how could we try to reduce this one? Perhaps maybe reduce the 25% maybe to around maybe below 5%, or reduce the 80% of potentially human pathogens to maybe 40 or 5, maybe below that. Here we have listed some of the modifiable factors, and I would not dwell on this one because they have been mentioned by the previous presenters. And on vector-borne, just to mention a few, on vector-borne, we have seen that one, especially on tick and mosquitoes, the expansion of their distribution, density also changing because of the temperature change and also rainfall. We have also seen the waterborne increasing because of the quality and the drought that comes. We have then the shared water resources with the animals, both the livestock and wildlife. And this one has basically led to increased cases of cholera, diarrhea, diseases, and other enteric problems. Airborne, as we have seen, because of the human population also increasing, the transport has caused one of that one to be a big thing, and also because of the change of the climate and weather, air quality, air pollution indoor has also exacerbated that situation. On zoonotic, we are talking about Ebola and Mpox, as mentioned by Stephanie, and because of this one, we have in Kenya we have quite a number of human-wildlife contact, bushmeat being consumed. And all these ones because of many things that are happening in the landscape. So trying to address this in our own situation at the One Health Center, we try to see how then all those comes together using the One Health lens. And here we are talking about the landscape we are living that is shared water resources, the environment that harbors the vectors, as we've seen in this picture. We have the animals, the wildlife, and we have the human beings where when we have food insecurity, then the possible poaching occurs, the bushmeat occurs. So in this pictorial, you are able to see a human there carrying a baboon or monkey as part of the food chain. And then as he walks along the environment, there are vectors, there are ticks, the mosquitoes, and also the vectors there, like the bats that also in the same situation. So trying to address this one, to speak to the same thing we are talking about the Lancet and the WHO, where the 25% of the human deaths are because of this one. If we're able to know what's lacking out there, then you can be able to institute a better kind of intervention and also a remedy to try and reduce these ones. So while you are doing some of the work, we have seen in some of the areas when we destroy the habitat because of that land use changes, when the drought exceeds. So when you destroy some of the forests and the trees where the birds are known to dwell, so what these birds do, they come to close to the man-made caves or maybe holes, as you see in the right picture here. And those caves are shared by other animals together with humans, as you see in the lower picture here. So those are animals that goes there maybe to seek their refuge. There's also some holes there inside there where people keep the honey. beehives were for honey. And so this kind of high interaction of both wildlife, livestock, and human kind of interface. And in such case, you find that potentially then you are speaking about multiple possible pathways of exposure and spillover of potential pathogens that these animals or vectors may be sharing. In one of the areas that we went to the field, that's some of the what happens when you destroy the forest or the habitat for these animals. These are bats basically roosting within the avocado tree close to the household. So from the avocado here, we're talking about maybe less than 5 meters to where the house is. So you can imagine the children playing under this tree. Some of the young ones may be getting somewhere those avocados there. The avocados will have some bites. And so we know that the saliva and the guano from the bats do contain quite a number of pathogens, potentially harmful to human, including the rabies virus. So when this one happens, then we are talking about high risk that could be able to then spill as from one index. So from one index case, from one index case, then you have the spillover to the rest of the community. To continue our bat surveillance, so we went to one of the dispensary in one of the northern Kenya. So these are dispensary in part of the country, in the northern part of the Kenya. And within 1 hour, when we put our mist net, we were able to capture a couple of thousand of these bats until our mist net just fell down. And all these bats were roosting at the ceiling of the dispensary. The unfortunate case that they were roosting on top of the ceiling, and under that ceiling is where the pharmacy is. We are talking about the syringes, the drugs, the needles. So you go into the dispensary for your normal kind of a medical check, and then you get swabbed by the gauze bandage that has been contaminated with the guano or the bat urine. And so you have this kind of infection that you did not go with, but you live with the potential of some of those infections. So these happen in the dispensary where the clinicians have no idea that potential risks that they're staying with. So when we collect the samples from those animals, from the bats, we collected the blood, the nasal swabs, the oral swabs. We're able to detect some novel viruses, including the coronaviruses, the astroviruses, and retroviruses. And some of those coronavirus, they were just maybe lacking 1 or 2 amino acids to basically potentially maybe jump the host from maybe where they are into the human. So we don't know what else maybe those bats they may be containing, yet they are roosting within the dispensary where people are going to seek healthcare. So just extending the same thing that's happening within the landscape, we have just spoken that as the animal moves, they move with the vectors. And so when we have either wildlife movement or maybe livestock movement, you have a number of millions of these vectors also being carried along the corridors with those animals. And so they're being dropped along the way. And so you may not have moved when you go for clinical history, you may not have moved from your area, your homestead, but the vector might have found you there. And that vector may have come maybe 200, 300 kilometers north, and it's maybe carrying some potential pathogens. And then when you get bitten with that one, so you get to have an infection of a pathogen that's not endemic to your region. So the clinician may not be able to detect that or identify that or relate with that one because it's very new, and something that is not aware because it's not endemic to the region. So the reason why we do this is because of the disconnect that we do have when we go to the health facilities and try to understand how the management of the individual cases of the population is being done. So some of the work that was done by one of my master students, she was able to find that there were quite a number of AFI or fever of unknown etiology that's being reported in the clinic. So an individual goes seeking medication because maybe is feeling maybe fatigued, has some migraine, is maybe having some diarrhea. So what the clinician does, basically will test for normal endemic diseases like malaria, typhoid, or brucella. And when those returns negative, the only thing then, and the individual is sick, then the person will just be recorded as fever of a known etiology. So what happens in such a case? You are given a broad spectrum antibiotic, and then maybe a week or maybe a few days down the line, then you relapse, you go back, and then you are given the same medication or a different medication. And then what happens that someone may be 2 or 3 times visiting the hospital without any response, then that person goes into herbal medication or self-medication. So we sampled the community there, and these were non-syndromic individuals, people who were not showing any clinical signs or not in the hospital, just normal health individuals from the community. And what we saw is the disconnect of what was being reported in the clinic and what the community were having. So what we saw here, an individual having a co-infection of multiple of these viruses. So there was one individual who had Crimean Congo, had RVF, had some flaviviruses, one person, and that person behaves normal, but when you check the clinical history, maybe 6 months down the line, he or she had reported some maybe severe headache, some pain in the ribs, chain, some coughing. But when that person into the hospital, was recorded as fever of a known etiology. So the hospital, the health facility was not able to detect that that person was suffering from this myriad of the viral infection. One, because of the limited lab capacity. Two, because some of them are not in the disease, some of the pathogens are not endemic to the region. And this is because of the movement of the animals and the vector expansion that's becoming now a problem. And so the diseases is kind of always expanding the horizon where it's found. So that disconnect leads to what then you are given medication, and medications that's not meant for actual diagnosis because there's a misdiagnosis, and then that leads to another problem. And that problem is what we're talking about now, the antimicrobial resistance. So what we do to address that one, so once we have the findings, we go back to the health facility. So on the left picture here is one of the health facility in Rumuruti. We are basically with some nurses around the region. We are trying to walk them through the findings, telling them what exactly is happening in their landscape and how maybe to address that one. How could they narrow their differential diagnosis and potentially maybe manage the community. But at the same time, we try to also talk to the community themselves on the right and try to understand their side of the story, the problem they encounter when they go to the health facility, whether maybe they're able to receive the right medication or the right management and all that. So that's the what we try to put in a triangle and try to address that one as a team from the One Health. So because of that misdiagnosis or failure to pick the right diagnosis, then you have antimicrobial resistance being reported. And unfortunately, new drugs are not being manufactured. We are still using drugs that were manufactured in 80s, early 70s. And so with the AMR coming, there's a threat. It's reported by 2050 that will be around 40 million deaths that will be due to AMR. And so these deaths will basically overcome or basically be more than what the infectious disease will be
Dr.
causing.
Kamau,
so if you think about...
Dr. Kamau, sorry, I need to interrupt you. Can I give you just 1 minute to wrap up?
Yeah.
Because our next speaker is already here.
Yes. Okay. Yeah. So this is what we normally have. And so with this kind of myriad of problem, we end up having the problem that we basically having. So because of that one, so the AMR that we've seen over the 40 million plus, the 40 million occurring by 2050, we need to address that one. And so some of the students have been trying to work on that one. The AMR, as we see here, we're still trying to see whether the landscape is also contributing to that. And we also basically seeing that there might be something that's happening from that and that we may need to address of what AMR is causing or is originating from. So we've been trying to reach the community through poster and different ways of educating them so that they can be able to respond. And in these pictures, we are talking to the community health facility, but also to the students who basically are the first index cases of the problem that we normally encounter. So to do that one, we have to have a prediction, as was mentioned by previous presenters, and how to do that one. Then we are going to overlay different data of the hazards, the climate, the transmission pathways, to be able to come up with a framework, as we're shown here. So we are trying to work on this framework that we can be able to identify the stage or where the spillover occurs, the interface that you can be able to do an intervention, and how whether that intervention will be able to make some positive changes to the community. And with that is the one team that you always in the field together, trying to make this happen. So I would want to thank you, Dr. Mercy, for trying to fast track my presentation by a minute, but also for agreeing to co-chair in my absence with Dr. Mwangi and all the presenters for the One Health session. Thank you for making this one possible. Over to you, Mas.
Thank you very much. Okay, so again, I'm standing here. I'm the one who's standing between you and the tea break, so I apologize for that. But we do have our keynote speaker who is already online. So the proposal is to have our keynote speaker for the next session, and then after that we'll break for tea break. I hope that will not induce stress to any one of you. Ruth, Dr. Ruth?
Mercy, I'm very happy to come back after the tea break. I'm not in a hurry at all.
Good evening, everyone. And as Mercy said, I think we're going to start in the next session, which is on research and development initiatives in reproductive health and infectious diseases. And this session, I'll co-chair it with Dr. Atunga. So for our first presentation, we'll have Professor Niroshini Nirmanlan. She completed her undergraduate degree at the Faculty of Medicine, University of Colombo, Sri Lanka, before earning her MSc and PhD at the University of Salford, Manchester. After postdoctoral training at UMIST and Kruk, she joined Salford as a senior lecturer in April 2010 and became a chair in 2017. At Salford, she has established 2 internationally recognized research themes: malaria drug discovery and biomarkers in major trauma. She leads the malaria research group, pioneering drug repositioning as a fast-track method for anti-malarial drug development, leveraging existing knowledge of drug bioavailability and safety. Professor Nimalan has built extensive collaborative networks with institutions like KEMRI and KIPRA in Kenya, NHS Manchester, Royal Infirmary, and GSK Tricanto. She has held various leadership roles, including program leadership and head of biomedicine. Additionally, including program leadership and head of biomedicine. Additionally, she serves on several committees, such as chair of the University Fitness to Practice Committee and external examiner for postgraduate medicine at Anglia Ruskin University. A staff elected member of the Senate of the University of Salford, that was between 2018 and 2025. She is also the Senate elect chair for the University-wide Academic Bureaucracy Review 2025. We welcome you, Professor, to give us your talk.
Can I ask if you can hear me?
Yes, we can hear you.
Okay. Will you be sharing my slides, or do you want me to share it?
Yes, you can share it afterwards. Thank you.
Okay, check. Can I
Yes, we
ask
can
if
now
you
see
can
your-
see my slides?
Yes, we can see your slides.
Okay, right. Good afternoon, all, and greetings from Manchester, very grey Manchester we have here. My name is Niroshini Nirmalan, and I'm the Director for Human and Natural Sciences at the University of Salford, as you just heard. I lead the malaria research group at the university, and we've had quite a lot of collaborations with the University of Nairobi and Kipre. And let me see okay. Yeah, so starting from somewhere in July 2023, we have made quite a few visits. The work we do kind of aligns well to the UN SDGs around malaria. And I've had many research trips there, including to places like Siamos and CIR and the Cambry centers as well. And of course, Kipre more recently, and collaborating with Lucia Chola and her team. I'll kind of give you, I'll eventually come to the work we have done and why, and the background there. But before that, let me just tell you a bit of a story about malaria, which I'm sure you know, but it's always a very fascinating topic to talk about. It predates humanity in the sense that, all kinds of mosquitoes in resin. There's evidence that there was a plasmodium-like infection at that point. You can think of the gut oocytes that have been preserved. But of course, there are also very ancient references to these drugs. And although you quite don't see the tertian and the quartan fevers anymore, I started life as a medical doctor, and I do remember the times where you have a fever chart by each patient and you look for this spike and in the massive hepatosplenomegaly and all that. Not quite the scene now, because people, we do treat them early, but it used to be quite a picture. Malaria and quinine, of course, have historical significance, probably the oldest drug prescribed. And if you talk of one drug that has had a hugely powerful effect, that has to be quinine, also known as the Jesuit powder, not because the Jesuits discovered it, but more because they kind of used it for malaria. It was used by the Incas in South America. When I do my lectures in Salford here, they are always very fascinated to hear that, even as late as the 1940s, malaria was not this distant disease that they think it is now. But even in places like Europe and America, it was a problem. And I wonder if you, one of the books I would recommend is Fiametta Rocco's book called The Miraculous Fever Tree, where they talk of the papal conclave in 1623, where there was a lot of competition, and at that point they didn't realize in Rome that malaria was caused by a mosquito. They thought it was bad air, so they sealed the Vatican. And of course, malaria takes hold. They couldn't come to a decision to elect a new pope. And by the time the new Pope Urban was elected, there were so many deaths within the conclave, and a very graphic picture is presented, including the new Pope himself, who the first thing he does when he gets well is to send missionaries far and wide to look for a cure for this dreadful disease. And Agostino Salambrino is one of the priests who go there and then find that the Incas are using cinchona, the bark of cinchona, and of course they brings it back and tries it on malaria, and the miracle happens and it works. All these names are thought to have either, succumbed or at least developed very serious illnesses, from Caravaggio to Byron to Genghis Khan, David Livingstone, of course. So it has been a disease that has, spread its claws far and wide. And I really like this. This is Albert Dürer, the artist, who sends a painting of himself to his physician, pointing at his spleen, basically telling him it hurts, it really hurts here. So the soft spleen in malaria is one of the characteristic things. Yes, so even at that point, he could actually co-locate his illness and send it to his physician. But of course, he died of the disease. The drug quinine, a natural product. If you saw my title, I'm talking of going back to our roots. When I mean roots, I'm also, in the widest context. Quinine was one of the most powerful drugs that has been used over the centuries. And of course, a lot of history around quinine itself, where quinine wasn't used, the Panama Canal, the Suez Canal projects. There's again a lot of history in that book, and I very much would recommend it. Colonization. I mean, what would colonization have been if not for quinine? A naive population of foreign soldiers coming in to the south and to the tropical countries ridden with malaria, and if not for quinine, maybe the landscape would have been very much more, the picture, history itself would have been very much more different. We have World War II, where General Douglas MacArthur says, this will be a very long war. If for every division I have fighting the enemy, I must count a second division in hospital with malaria and a third division convalescing from this debilitating disease. So yes, I mean, mother of all diseases. And if there was one drug that had impact on humanity and history, it has to be quinine. What's the situation now? Many centuries later, it still has its grip. Deaths are not in the millions, but it's just less than a million, 600, 700,000. That's still so much more, far too much. And with all our modern interventions, we still have this picture still. The picture has moved down to the global south. I really like this image because if you see at the bottom of India, there's a little island, and that's where I come from, that's Sri Lanka. And that turned green a few years ago. Malaria was eradicated from Sri Lanka. So absolutely proud of that. But of course, drug resistance is continuing and spreading, and we have to start thinking how to get over this issue, because if you know the life cycle of malaria, it is quite a complex life cycle, and that is probably part of the reason why interventions are so difficult to find as well. Control, yes, you have the vector control, the environmental control, the routine things that are done. Vaccines, that's the hope. I don't think anything spectacular has really happened there, but the mainstay of antimalarial control has continued to be chemotherapy. And if you look at the big groups, the quinolones, the artemisinin, the modern ones, and antifolates. Antifolate, the lifespan has been relatively short. But if you look at quinolones and artemisinin, they're both natural products. Maybe there's a lesson for us there. Drug resistance, drug discovery in the 21st century. The number of new clinical entities that are being found are diminishing. And R&D costs are huge. If you look at the traditional drug discovery pipeline, it takes years. And this is not good news. Regulatory control is increased, health and safety, there's a bit of an overkill there. Someone was saying recently, I listened to a talk, they said even paracetamol won't go through the current regulatory control. And of course, the traditional big institutions, industry, academia, regulatory bodies, there's a mismatch. There's no communication. Each one works at a different pace. Patents, profits, high throughput technology. This is another issue. We are over reliant on this. We thought there'll be a magic bullet when omics came, and you chuck something in, and the other end you'll get the result. That was not the case, and because of that, we deprioritized natural product research. Right, for a long time, natural product research, nobody wanted to get their hands dirty with that. So therefore, the drug discovery pipeline is pretty bleak for malaria. Of course, this is Professor Yu too, and if you look at she has spent a lifetime looking at traditional Chinese medicines over 40 years or 50 years or something like that, screening those after the Vietnam War. And then what we had was a product, this chemical entity, artemisinin, with that double bond inside the ring, which was not even known to man. So you can see what over time, with evolutionary advantages, how these molecules are developed. And this is artemisinin, but of course the bad news is even artemisinin is now you can see an end to its use. And we just cannot go through this long drug discovery pipeline because we may really not have the time. So one of the things that we did, our group in Salford did, was to look at drug repositioning. So what is drug repositioning? Is to look for new users for old drugs. We have a whole range of old drugs which are used for a variety of things. So the idea was to take these drug libraries and screen them for malaria. And we use the low-pack libraries, which are patent-free, and also the enzo drug libraries. About nearly 2,000 compounds we screened. Just I'm very quickly going through. This is what a typical screening panel would look like, and it was a single 2 micromolar hit with the drug, and it was all blinded, of course. And there were when the blinding was kind of taken out, we could see that our methodology was nice because there were also antimalarials, and you can see that the antimalarials were quite nicely suppressed. So to cut a long story short, so we had developed a cybergreen methodology. It's there in our first paper, I think. It is not a high throughput screening. It's a normal 96-well screening, but it was a moderate throughput screening technique. And to cut a long story short, we got quite a few hits, at least top 50 potential hits from different candidates. Some of them were the existing malarials, antimalarials, antibiotics, there were anti-cancer drugs, antipsychotic drugs, and a whole range of calcium channel blockers, ACE inhibitors, and so on. So very interesting profiles. The story today, I'm going to just tell you about one drug which was pretty high on the list, and the drug is called emetine hydrochloride. Those of you who are closer to my age will kind of remember the drug, but most people in the West, it's something not heard of. Emetine was used in amoebiasis, and in the 70s and 80s, Amoebiasis, of course, was rampant in Sri Lanka and India. I'm not sure what the situation was in Kenya, but most clinicians would have been very, very familiar with amoebiasis. You had gastrointestinal dysentery-like presentation, and then you had a hepatic abscess, which is very, very awful, and you had to use very high doses of these drugs. I mean, this drug was, available over the counter. Kids with amoebiasis were they had this treatment and so on. So yes, it was a commonly used drug. It wasn't without side effects, right? The word emetine itself must tell you, emetine is emesis. Emesis is vomiting. So it was not a pleasant drug, and that was, vomiting was part of that. And there were also, when the very high doses were used, there was some cardiotoxicity. But the cardiotoxicity there really was ECG changes. So when high doses were used for hepatic amoebiasis, the recommendation was that you take an ECG and check that these changes are not visible. But what we were seeing is that the IC50, so this is nanomolar, 47 nanomolar, so it was highly, highly potent. And because I had this memory as a medical student, in Sri Lanka, I was absolutely thrilled because I knew this was used, and I thought, this is great, and I kind of started looking at it, and I got my PhD student, there was Holly there, to look into this drug and let's start working on it. And there are lots of tick boxes that need to be used. Now, in the 1980s, the use of emetine was stopped, it was banned, but metronidazole came into being. And metronidazole was a much more pleasant drug. So kind of mid-70s and 80s, the use of emetine kind of stopped. And if you look at the literature after the 1980s, you really don't see anything much about it. It kind of died down because everyone forgot about emetine. And nothing was done. It wasn't used. And metronidazole was a lovely drug, and it was used for amoebiasis. Still not as effective as emetine, but that was what was used. And amoebiasis went down as well because of the health education that went with it. So alongside us, there was another group that actually, in the Liverpool School of Tropical Medicine, there was another group, Lukumi et al., who had also done the screening. In fact, they had better nanomolar values. However, for some reason, they hadn't taken, when they progressed to drug leads, they really didn't take this drug forward. So, which was kind of surprising why they missed that. But I thought, let's work on it. No one else is. So, as you know, there are these WHO gold standards, the parasite killing rates and PCT, PRR, and all that. So we did all that and wrote the paper. Everything was looking really nice. We sent the paper, and I think it was Malaria Journal, and what we got was an outright rejection. Very, very strong rejection. And the editor, who at that time I think happened to be from the Liverpool School itself, the reviewers were scathing. They were almost saying, what is wrong? What is wrong with you? Because you're suggesting a poison. And the word poison kept, and I was stunned because I was wondering what on earth is happening. So then we went back. We were very bitterly disappointed. We were putting in so much work on this drug lead to optimize it, and now this. Then we went back. That was the first time I kind of went back into the literature to see what had happened. And the simple emetine safety data sheet, if you looked at the emetine data sheet, if this is the main document that kind of defines a chemical. And there it was, fatal if swallowed, right? They had condemned this drug, which was used by thousands of people, probably millions of people over a long period of time. And it said fatal if swallowed, and it said if swallowed, immediately call the poison center. So this was kind of shocking. And then we went on to look more at it. And then I don't know whether you remember this lady. This is Karen Carpenter, one of the famous singing duo, brother and sister duo, the Carpenters. Now, Karen Carpenter suffered from bulimia. At that stage, of course, bulimia was very new, where you would induce vomiting and all that for weight loss purposes. She was using emetine. She was using the syrup of ipecac, which kind of which did have emetine, but also had other extracts of the emetine of the ipecac tree. And what happened was then she did overdose, and she died of emetine. And the fan club then lobbied the FDA to ban this drug. So that is how it all kind of changed, and the impression changed. So I had to then come up with some very another reason, and that was, of course, side effects are dependent on dose, and only dose makes poison. So I also looked in at the previous data, looked for the amoebiasis data, and funnily enough, the IC50 for amoebiasis, the IC50 needed, comparative IC50 needed to kill the amoebic parasite was micromolar, 26 micromolar, so almost 1,000-fold higher. So we are talking of a different dose range. And if we go by, and the side effects are dose dependent. So I had to write back another strong letter to the editor and kind of explain this and say, look, you cannot throw this because you're talking of a whole different dose range. And if you ignore this, we might be, it's so effective. And funnily enough, he wrote back saying, I normally don't reverse my decisions, but in this case, I do see a point. And we had our paper published for the first time with emetine, which is a great thing for us, because after a long time, after many years, again, emetine comes back into the story, into the picture. And of course, emetine is a plant-based plant product. So side effects, I'm going to just quickly go through this.
Can you hear me?
How do we overcome side effects? So we know that emetine, the two main side effects for emetine is nausea, but with current formulations, with coating and slow release preparations, nausea and vomiting are not big side effects. You can even add a second antiemetic with the preparation and so on. The bigger side effect, of course, was the cardiotoxicity, and could we chemically change this molecule? Can we chemically derivatize the parent molecule?
Hello?
Could we look at synergistic partnerships, which will further reduce
Can you hear
dose?
me?
I'm sorry.
Hello, can you hear me? We are running out of time. I can give you 5
Okay.
more minutes to finish up.
Okay, right. I will. Right. So okay. Yeah. So what then also happened was that in Imperial College, after our publication, after our work, also came up with the binding site of emetine and found the target binding site. So which meant that we knew where emetine was now binding, so we could actually change the molecule and derivatize the molecule and look for analogs with less side effects. And this was work done. I'm going to just go through. This was work done by another PhD student. We designed 2 diastereomers, 2,3-dihydroemetine, which when tested, we found retained. So these are diastereomers that will not, which are excreted faster. We were not the very first people to find it out, because GSK was already working on it. GSK was already in the 1960s. So we were able to kind of work on that and use the models, the modeling binding site that was that Imperial College had found to further refine, and these two products are now available, and that is forming the new collaboration we are going to have with or the ongoing collaboration we are having with Kipre. So it takes us to an exciting new phase. We already tested it on gametocytes, so it is gametocyte active. There is no cross-strain cross-reactivity, so it can be transmission blocking, and it also acts on the trophocytes, rings, and schizons. So it looks a really good molecule to be working on. We have now got ODA funding, and I work with Professor Jared Mecha and Dr. Lucia Chola. So we are going, that's the next phase of the project, and we're going to hopefully work on the mouse models and then on the non-human human primates to look for efficacy of this molecule. So watch this space, and thank you very much for hosting me. And I look forward to continuing the collaboration with Kipre. Thank you.
Thank you very much, Professor. And that was quite exciting, the whole issue of drug repurposing, and we are really looking forward for the research. I think for now, if for any questions, I think we can be able to reach her. Probably we can reach you maybe online.
Yes, please, just share my email, and I can, Dr. Lucia Chella will have my email, and I would very much like to collaborate, have the conversation. Thank you.
Thank you very much.
Thank you. Thank you very much.
So for now, I think we'll go for tea. I think we'll just take a 20-minute break for tea, then we'll come back for the last session. Just to give you an incentive, after this session, there are cocktails. You'll be invited for cocktails. So once you take your tea, I hope you'll come back rejuvenated. So maybe we can take a break for now for 20 minutes. We'll be back at 5:20. Department of the Tropical and Infectious Diseases at the Kenya Institute of Primate Research. Karibu, Daktari.
Just give me a minute as they load the talk. Okay, so let me get started. The challenge with me standing here as a head of department, my team feel they can slash down my time. So they only got to allocate for me 10 minutes, but I'll do my best. So good afternoon to you all, and thank you very much for this opportunity. So this afternoon, I'm just going to talk about the microbial Jekyll and Hyde, exploring the dual nature of Schistosoma mansoni. In our department, just quickly my outline, I'll be discussing, it's very difficult when you have to look behind. Basically, I'll just introduce the department that I head, just highlighting some of the programs that we have. And then I'll go into the whole schistosomiasis research program that we're doing, highlight some of the studies, and just bring to, it's actually dark, unfortunately. So maybe you can sort that screen in front, it would help us on the podium here. So I'll then introduce schistosomiasis, highlighting some of the programs that we're doing under that program, and then just have a final conclusion. So this slide is not new to you, but I just wanted to highlight quickly the infectious diseases program started in the early 1980s. We have many research programs that we do. There's malaria, there's virology, there's the NTDs, and we also host the biobank facility. And what would be of interest is the schistosomiasis program. For myself as a research scientist, I work within both the malaria and the schistosomiasis program. Into the background of schistosomiasis, it's an ancient disease. It still continues to plague over 250 million people worldwide. In terms of how it presents clinically, you have the acute disease that develops around 4 weeks post-infection, and if left untreated, it becomes a chronic disease. In terms of the life cycle, we have 2 stages. We have the stage within the snails found in the environment. So the snail is able to release secretae into the infected waters. The secretae penetrate the skin, enter into the body. You have the secretae forming schistosomula, which then can migrate to the portal vein and portal vein in the liver and mature into adults. So you have both the male and female fusing together. These paired adult worms will migrate to the gut and the rectum, and from there, the pair will be able to lay thousands of eggs. And as they lay eggs, they again defecate into water, and the cycle continues. But what is interesting to us in this field of schistosomiasis research is just trying to dissect the mechanisms behind how the eggs are able to transverse out of the liver into the gut and out into the environment. So that cycle has been well described, and I've just highlighted what it looks like. So the critical thing there is the eggs can become lodged in the liver and form these granulomas. In terms of the control measures, there have been many. We understand the ecology around it. We've had preventive chemotherapy focused mainly on praziquantel, so there's only one drug that was discovered in the early 70s. And the challenge with that is we still find reinfection happening, so that in terms of control remains a challenge. We've had people doing vector control, so if I come to Kenya and focus on the Mwea region, which is central Kenya, there's been introduction of a predator snail, which indeed eats the snails, but the challenge there is also destroying the rice paddy fields, so that can have some economic challenges in the long run. There have been other interventions such as improving water, sanitation, and hygiene, but for communities where they don't have access to clean water, that still is a tall order. We have teams working on vaccines. I don't have time to go into that, and there's a promising one that is still in clinical trials under the IVI program. And from what I've heard, this could be something that we may become available soon. But even as we have all these control methods, we now are looking at possibly a hybrid forming. So this is Schistosoma mansoni combining together with Schistosoma bovis. So these are current studies that are ongoing to assess what impact this might have, and I'd already mentioned the reinfections. We in the lab are interested in the immunopathogenesis. It has been well described in humans, but out of work done in our lab, we've been able to establish a model for this disease, the olive baboon, and I think Professor Yan mentioned that in his presentation this morning. But I just want you to appreciate that as we look at the disease manifestation, the progression in humans, and even in olive baboons, in the acute disease, we know what's happening immunologically, and as it becomes chronic, it shifts from a Th1 to a Th2 response. So this slide just highlights that, and just a few publications that have come out from our lab. But when I look at the good and the bad of what I've termed here as Jekyll and Hyde, we have some responses that might be protective, and there's some that could also harm the host. So when we understand this whole process, we can take advantage of that in several ways, and I think my talk will hopefully highlight what we're doing in that space. So into the model itself, we had established the olive baboon over 40 years ago as a good model for studying Schistosoma mansoni. In the field, especially in the Kibwezi area, you actually have natural infections that do occur. And when they established this model, you find that it's clinically, immunologically, and the pathological outcomes are similar to those in humans. And from previous studies from teams here at Cape Raven before I joined, they were able to do multiple treatment regimens to make sure that even as you infect, if you treat it parasite control, does it clear the parasite, yes or no? And even at Kipre, we're able to maintain the life cycle. So we actually have olive baboons that have these parasites in them. And what we use them for is passaging through the snail, and then we can do studies in both mice and other olive baboons. So what I want to highlight quickly is just some work that has been ongoing in our lab. The first one is looking at what impact does schistosomiasis have on vaccine responses. So both Ruth and I had a student, or still have a student, Linda, who's just winding up and hoping to defend, but she will have a poster from tomorrow, so I'll just invite you to go and look at that poster. So here what we are looking at is the impact of the presence of schistosomiasis and what it does to vaccines such as human papillomavirus vaccine. The other work we are doing is, and Ruth is going to present on this, but mine was just to highlight that we've been looking at the improving diagnosis. We still use the gold standard microscopy, so Ruth will highlight this. She's only giving me 5 minutes. We've also got a study where we're trying to dissect the mechanisms around egg migration. But for the rest of my talk, I just want to share with you some data that we got from a study. We know that Schistosoma mansoni is able to modulate the immune response, and sometimes that immune response, the Th2, may favor some autoimmune diseases. So we decided to do a small study with the master students from Pan-African University. So here what we know is that type 1 diabetes is able to destroy pancreatic B cells. And some of the driving mechanisms behind this is that if you have interferon gamma present, it can promote autoimmune inflammation, pancreatic B cell destruction. So what we decided to do was evaluate some of this, the peptides that are released by Schistosoma mansoni. So evidence is there already to show that schistosoma egg antigen can somehow confer protection against diabetes. So in our lab, we then, with the support from the Mount Kenya team, we were able to produce these peptides, which I've written, I'll show you in the slides, iPSC1 and omega-1. And we were able to test it in a mice model, in a rat model induced with diabetes. Just quickly to the methods. So the peptides were generated from molecular docking. We looked at the structures and ensured they were able to recognize the site to which the peptides can bind to the actual antigen sites. We induced diabetes in our rats at that concentration. We did several interventions. We had a control of PBS. We used insulin, which is standardly used for treatment of diabetes and management. And our intervention were the SEA. So this was just a quick pilot study. We're able to treat for 5 days and then monitor the glucose outcomes. In terms of what we saw when you look at blood glucose levels in these rats, on your left, I had the control of PBS. We had insulin, and 1 day post-treatment, if I can just, I think something jumped here. Okay, so in the insulin arm, 1 day post-treatment, we found that the levels of glucose declined. And if you compare with the peptides that we tested, the crude SEA antigen, together with iPSC and omega-1, there was also a decline in the glucose levels. We're appreciating that this was such a short space of time to test impact, but we're hoping now to recruit a PhD student just to extend this work and ensure that the results we found are genuine. So that's something that is currently being worked on. In terms of cyclotime levels, I'm just highlighting there that, again, on the 1 day post-treatment, we were able to see with SEA, omega-1, and iPSC, an overall reduction in the IL-4 cytokine. So from us, with this preliminary data, just to move on quickly, we can just say that it seems to have an impact, the soluble egg antigen, the peptides that we found. And if you look at it histologically, on my immediate left on the top there is a healthy, normal liver that we were able to take excise from the mice, the rats that were given the treatment. And if you have the PBS, you can see a lot of infiltration of white blood cells. So that's the one in the middle up there. And when you do omega-1 and the different peptides from the schistosoma parasite, you see some moderation in infiltration of these white blood cells in the liver. So overall, what we can conclude there is that helminth-derived proteins can actually modulate autoimmune diseases like type 1 diabetes. From the peptides we studied, S-mansenite peptides presence do reduce the pro-inflammatory interferon gamma and it seem to enhance protective IL-4. And what we are looking at now is trying to extend this work, maybe do a bit more detailed study to ensure that the results we find are bona fide and good. And most importantly, we are trying to drive into product development. So I'm hoping, and this is me just telling my boss there, that we're hoping this could be something, a candidate molecule that we take just beyond the mice work, the rodent studies that we do. So with that, let me just quickly draw to a close, just to say that overall in our lab, the olive model has been a valuable model. We have not stopped at just testing vaccines. We've looked at improving diagnosis, and we've moved even into the molecular space, and Ruth, I hope you'll highlight some of that tomorrow. And then as scientists, we have to be creative. So we're trying to take advantage of this particular parasite to then generate possible peptides for treatment. And lastly, we hope we can translate some of our findings and take it further into human work. So with that, let me say thank you to the team, an extensive team. I can't take the credit. I joined the program when it was already advanced. So to all my schistosomiasis group, both new and old, they still continue to contribute, and to the different teams that I work with. And here I'm highlighting not only my schisto, but also the malaria team that support the different work that we do. So with that, I say thank you and hand it back to the chair.
Thank you, Lucy, for keeping time. Now we're going to switch gears. We've talked about tropical diseases. Now we'll go into the space of reproductive health. Our first speaker will be Professor Tony Perry, who is a professor of mammalian molecular embryology at the University of Bath, England. You're welcome to give your talk.
Thank you very much. I'm thrilled to be here, and if I can get the presentation up, there we go. What I'm going to talk about today is using sort of nascent technology, lab-based technology, as potential approaches to conserve endangered species, particularly non-human primates. And in this talk, I'm going to start by talking about how we might move bits of cells around. And then for the most of the talk, I'll talk about how we might move whole embryonic cells around. So the first part is really about nuclear transfer cloning. And so in nuclear transfer cloning, here's a schematic that summarizes what we do. We take an egg, a mammalian egg, and we enucleate it, so we remove the chromosomes. And then we take chromosomes from the individual that we want to clone, the nucleus donor, and we inject them into that enucleated oocyte and then cause development to begin. And then the developing embryo is transferred into a surrogate mother who carries the pregnancy to term, and a cloned individual is born. Now, what we might do here is a variation on this, where we're actually using different species. So the oocyte donor is a baboon, because it's relatively, we think that baboon oocytes will be relatively available to us. And I'm going to use the example, perhaps overused example, of the Tana River mangabe for this talk for the endangered species. So we want to take then a somatic cell, a nucleus from a Tana River mangabe, and inject it into the enucleated baboon oocyte, cause development to begin, and then transfer the embryo into a baboon who will carry the pregnancy to term, and then we would end up with a cloned mangabe born. So that's potentially might help us to propagate the mangabe. So there are precedents for this kind of heterospecific nuclear transfer, dating back over 2 decades now. Here's some of them, and for example, the gaur, which is an endangered large ox species, has been used as a nucleus donor into cattle, regular cattle oocytes that have been enucleated with the birth of live offspring. But there are considerations in all of this that I'd like to consider with reference to de-extinction, because there's a parallel track being taken by the guys who want to resurrect some species, for example, the woolly mammoth. And it seems like hardly a day goes by without there being a woolly mammoth story in the press, at least in the UK. So here are some considerations that we might raise in parallel, considering what they're doing in the woolly mammoth. And the first is that they need a recipient oocyte and host pregnancy in the closest tractable relative. And in this case, it's the Asian elephant. And the Asian elephant, because we have genome sequences from woolly mammoths and Asian elephants, of course, they're very similar. They're relatively similar in genome sequence terms. And their nuclear mitochondrial compatibility, there might be issues there. I think they're generally overblown, but there might be issues there. And we need to think also about whether the fetus, because if you do this nuclear transfer, then you're going to have an Asian elephant carrying a pregnancy of a mammoth. So it will give birth to a mammoth if all of this works. Now, if we transpose this sort of onto, well, let's think about Asian elephants in terms of what's known about Asian elephants. And really, if we go through all of this list of relevant techniques and things that we're going to need to sort of accomplish in order to get there, to get to our end goal of a clone, we really don't know very much. There are no ICSI, intracytoplasmic sperm injection Asian elephants, and we don't know very much really about their reproductive biology. So even the currently sort of living organism, the close relative to the mammoth, we know very, very little about it. And if we project from this onto, say, the baboon and the Tana River mangabe, what's known about art in the assisted reproductive technologies in the baboon, well, it's similarly lamentable, I'm afraid. So we've got a long way to go. And I think at Kipre, we can start to make serious inroads into some of these issues, which we're going to have to skittle down. But there are issues, and so we're going to have to think about alternatives, and I want to now go on to the second class for the most of this talk, which is developmental complementation. And in developmental complementation, the basic idea is that we take an embryo from a species of low concern, the baboon, and we mix it with an embryo from our endangered species, and they will integrate. The development will integrate. So what you have is a chimera, and when it's born, it's chimeric. I've shown it here. It looks like a baboon, but it's chimeric, and you hope that the endangered species embryo has contributed to the germline, the gametes, in this chimera, so that you can produce offspring. So the problem about this really, so there's one, there are actually precedents for this. I forgot to mention that there's a precedent, particularly high profile precedent from over 40 years ago, published by Steen Willitson and his co-workers, actually with this goal in mind, where he mixed goat and sheep embryos and produced something he called a geep, right? So it's successful, this thing was put on the ground, so it looks like this approach might succeed. The trouble with it really, that's a picture of Steen, the trouble with it really is that for Tana River mangabes, we probably won't be able to get embryos. If we could get embryos, probably it wouldn't be an endangered species. And so to get around that problem, we might be able to use pluripotent stem cells. So in this case, what we do is we take our embryo from a baboon, we mix them with pluripotent stem cells from the Tana River mangabe, they will integrate into development, and again, the same thing, we transfer the embryo and we hope that the offspring produce gametes that are derived from the mangabe. What are pluripotent stem cells? Well, these are cells that have almost the entire range of commitment options open to them. So they're cells that you can culture in a dish, and they can become any cell type that's in your body. And there are different types of pluripotent stem cells, and the ones that we're interested in are called naive pluripotent stem cells, PSCs. And these can give rise not only to all of the cells, but also to the gametes. So they could give rise to sperm and egg. So there are two types of PSC that you might have heard of. One is the embryonic stem cell, and that they are derived from embryos. And the other we were shown how to make in 2006 by Shinya Yamanaka. And he found that if you took somatic cells, so body cells like skin fibroblasts or buccal epithelial cells, and you zapped transcription factors from embryos into them, if you got the right transcription factors, some of those cells would become embryo-like cells, pluripotent stem cells. And this is called induced pluripotency. So kind of now we're potentially off to the races, because you can imagine we can get somatic cells from a Tana River mangabe without doing too much damage to the mangabe. And recently it's been found, this is in the last year or two, that there are really very, very powerful methods of doing this reprogramming to a naive state of pluripotency from in somatic, starting with somatic cells in things called, one's called super SOCs, and polycomb repressor complex inhibitors, and there are others. So it looks like we're off to the races in terms of making induced pluripotent stem cells from mangabes, at least potentially. So what we would do in this case is we would take our somatic cells, we would culture them in a dish and generate, this is from mangabe, and then generate iPSCs from them, and this is a colony, what a colony might look like there. This is the red stain is for something, I don't know what it is, probably alkaline phosphatase. They're adherent culture, then you can treat them so that they float in the medium, so that it's a suspension medium, then you can pick them up with a needle and inject them. This is showing injection into of pluripotent stem cells into the cavity of a blastocyst, so there's a pre-implantation embryo. And then you can take that embryo and you can, the embryo is a baboon embryo, and you can, with these now mangabe PSCs, and transfer the ensemble into a surrogate mother who will carry the pregnancy, hopefully to term, and then you'll end up with a chimera that I've called here a mangaboon. So you've got a mangaboon, and you hope that the germline has come from the mangabe. So that's one way you might be able to generate recapitulate the sort of germline genesis. But there are other approaches using related material that I just want to sort of, in the last part of this talk about, because these are exciting new developments, including in vitro development, where we can cause induced pluripotent stem cells to undergo the gametogenesis program, including meiosis. It's absolutely amazing in vitro. And so these are functional germ cells from male and female that have been shown to be functional in mice. In human, not yet, so they're still working on it. It's a very difficult problem, but there's progress, it's slow progress, and there's every hope that it'd be possible with iPSCs from mangabes. Also, and in a slightly different way, this is an illustration of embryogenesis in vitro in macaques starting admittedly at the moment with fertilization-derived embryos, in which these embryos develop for 25 days. So they've undergone somatogenesis, limb bud formation, they've closed the neural tube, and they have undergone lots of differentiation processes of a fairly advanced embryo, including primordial germ cell formation. So you have the potential of getting gametes from these. So is this relevant to bioconservation? Well, I don't know, but if it is, it might be via embryoids. And I want to spend a little bit of time talking about embryoids. What you can do is you can aggregate naive pluripotent stem cells, and if you do this in the right conditions, they will generate what looks like an embryo. So remember, these iPSCs are starting from an ear punch or a buccal epithelial cell, and you've generated them. So now you're generating embryo-like structures from cells that are originated from, for example, fibroblasts, potentially. And this technology, or this method, was developed by a guy called Nicolas Rivron, who I'll come back to, and published in Nature in 2018 for mouse and 2022 for human. And this is a kind of schematic. I'll skip through this in the interest of time. So it's derived from somatic cells originally. This is a key thing here in the context of our conversation. It's scalable, so you can generate many hundreds or even thousands of these things. So you might be able to pick one day the right one, the one that's most likely to continue to differentiate. And you can put these in microfluidic systems, and here's an example of this here, so that you can add factors to different sides of the embryo to break the symmetry and cause the embryo to start differentiating in the way that it would differentiate if it were in vivo. And so this is a quick movie that doesn't show it very well, actually, but it shows it a little bit of, if you look at the top left corner there, you can see the microfluidic structure. And then this movie, when I click here, hopefully it will start, and you'll start to see these embryos start to expand. And you can see there's a cavity in the middle, and that cavity resembles the blastocele of a blastocyst that would occur naturally in a fertilization-derived embryo. So putting this all together for non-human primate bioconservation, we're starting, of course, with our endangered NHP, and we have our low concern NHP, which is potentially a close relative. And the place to start, of course, will be to try and collect gametes from the endangered NHP and generate offspring in the way that you do. You can either use in vitro fertilization, IVF, or ICSI, intracytoplasmic sperm injection, and obtain offspring. But this, I think, is highly unlikely for the reasons I mentioned earlier. If you could easily obtain gametes, then we'd have to call it something else. It's not really an endangered species if they're that readily available. So we have to turn to some other process. This is just the summary slide of what I've just been talking about, really. I'm going over it again. But if you take an ear punch biopsy and you can do cell culture with those cells, you can use them in nuclear transfer cloning by moving the nucleus from the Tana of the cell culture into enucleated oocytes from the baboon, and you can obtain cloned offspring by nuclear transfer that way. Or you can generate low concern embryos, and using the other schematic, if you've got IVF and you've generated from Mangabe embryos, you can mix them together and produce offspring that way. But again, as I said, it's highly unlikely to be embryos available from the Tana River Mangabe. So we need a different approach, and I've suggested that we might be able to use naive iPS cells that you've generated from the cultured fibroblasts from the Mangabe. And with these naive pluripotent stem cells, you can direct gametogenesis in vitro and then do IVF and generate offspring that way. Or you can use the iPS cells from Mangabe and mix them with embryos from the species of less concern, the baboon, and generate chimeras that have germline from the Mangabe that way. Or you can take the naive iPSCs and generate embryoids and get development in vitro and maybe make gametes in vitro and blah, blah, blah. So as you can see, it's complex, and you're probably highly confused, as am I. So I'd like to summarize this, the last couple of slides here or so. I think what we can say about all of this complexity, there are lots of these different integrating technologies potentially, is firstly that there are multiple lab-based pathways that we might be able potentially to use to apply to conserve NHPs, particularly endangered NHPs. I'd also like to say that all of this, though, will involve expertise in NHP reproduction, and what you have here is that expertise, both under the stewardship of Peter Muedera as the DG of Kipre, and the team he's assembled, including Joram Karundi, who's speaking after me, and his colleagues and you. So this is absolutely pivotal to actually, what I'm saying might be incredibly fanciful, but the only way we're going to be able to do it is with a team of experts that are here. So that's fantastic, and it means that there's a golden opportunity at Kipre to realize these things. With the aim of realizing this vision of NHP bioconservation and embryoid research, and we are assembling a team as well, hopefully, that will interact. This is part of your collaborative ethos. The team that I'm establishing from left to right here is Brian Lamb, who's a Cambridge bioinformatician, computer scientist, biochemist. He's an amazing part of the team. Next to him is Martin Lieb, who's done a lot of pioneering work on PSCs in different species, including monkey species, actually. Next to him is Nicolas Rivron, this guy who's developed the embryoid methodology in the first place. And next to him, Shari Mackens, who's a Belgian clinician who's part of the team. We're very interested in, as you are, in the translational aspects of what we're doing. So some of these other aspects, though, that I'm talking about, so if you can develop these methods for bioconservation, they will also be usable for other things that may be of interest to lots of people. And some of these, this is not kind of an exhaustive list, include baboon genome editing. If we can actually get the reproductive biology and the micromanipulation working, then we can actually edit the genome to make models for post-genomic human disease, if we want to do that. Xenotransplantation, which requires genome editing. Vaccine R&D, you guys know much more about this than me. Embryoid drug discovery, so there might be applications for this, and also machine interactions. This is something which is quite controversial, but potentially very interesting in the future in lots of different ways. But if we return at the end of this talk now to the question that we asked right at the beginning, is all of this believable, or is it simply bonkers? Then I think what I'd say is this. First of all, it's admittedly speculative. The pieces, though, are in place, and we have an opportunity to use them. So I don't think it's bonkers. And indeed, what I do think is that it's not only not bonkers, but we can't really afford not to pursue it. And so I really commend the Kipre and the people who are you guys, and the people who are supporting you in your government to really take this opportunity and run with it before somebody else does. And with that, I'll thank you very much.
Thank you very much, and for also keeping time. Our next talk will be from Dr. Joram Kirundi, who is a veterinarian and specialist in reproductive biology and notobiotics from the University of Bern, Switzerland. You're welcome for your talk.
One of the advantages of speaking after Tony is that you can copy some of the stage mannerisms. So I'll take the microphone with me. So that's my name and my affiliations. And I will be speaking about the mouse. But to begin with, let us look at how important is the mouse. And for one who looks at even the recently concluded Nobel Peace Prize, of all the Nobel Peace Prizes, you will see that 102 of them had animals as a model. And out of those, the rodents took 94 of the Nobel Peace Prizes. And out of those 94, about 50% of them were mice. That is what you see as 43. And so the mouse will continue playing a significant role in human disease models. But Where does the story begin? This is a kind of a chicken and egg question, but I choose to begin in the uterus. In the uterus, the offspring is considered to be in a sterile environment, or so it has been for over 100 years, until recently when that paradigm was challenged, and someone argued, or a group of researchers argued, that there are microbes in the uterus. But be that as it may, the amount of microbes that we have in the uterus are not sufficient to mount a significant immune information or immune education, and therefore it can be considered still very sterile environment. And depending on the route of passage, it is at birth that the offspring is going to acquire microbiota. If it goes through the vagina, then it will be vaginal microbiota that it acquires. If it is through cesarean section, it is the skin microbiota that it is going to acquire. This microbiota is irreversible. It stays on, and that is us or animals. And so we also know that the significant mucosal surfaces are occupied by microbiota, be it the skin or the linings, the gut linings that we have. Most of the microbiota is going to be in the gut, as you can see, but all other surfaces do have. And we can also see, if you are to think of the microbes vis-a-vis the eukaryotic cells, it's almost an equal number of, in terms of ratio. The microbiota are prevented from going into some spaces because of the barriers, the mucosal barriers. But the metabolites from the microbiota permeate through these barriers and go into every part of the body. And so one can see through the dotted lines here that every part of the body from the brain to the toe has access to these metabolites and therefore will be influenced in one way or the other. One can therefore argue that we are half human and half microbiota in terms of either genes or cell ratio. But why is it important for us to think about microbiota? And this is where I will borrow Dr. Chola's phrase, Jekyll and Hyde, that it is because we have a Jekyll and Hyde phenomenon here. The microbiota influence most of our physiological systems and also some of our developmental systems, from the development of the blood vessels into breaking down of food particles and many other of the healthy physiological systems. But that is the Jekyll of the paradigm. We have also a gazillion of problems in that most of many of the diseases are associated with microbiota from the gut and from the skin. And so all these microbiota that we have will fall in one of those categories. Either it is a healthy microbiota or one that can eventually lead to disease. So the problem, as we said, is that when you get associated with the microbiota, there is no way of separating yourself from the microbiota. And this means it is very, very difficult for us to be able to determine which microbiota eventually cause disease and which one is a healthy one to retain. Except that in the last 100 years or so, there has been this germ-free mouse model and many other animal types models. We have had even non-human primate models where you can be able to separate colonization from the host. And that is through assisted reproductive technology. Here what I'm showing is we have a colonized donor of the oocytes, and we can be able to superovulate this female, and principally what we do is inject with follicle-stimulating hormone, say on a Monday, and then on a Wednesday we shall stimulate with a luteinizing hormone type injection, and then 1.5 days after we shall get 2 cell stage embryos. These 2 cell stage embryos now can be transferred into a timed recipient. And for a timed recipient, you want to use the Whitton's phenomenon, where you mate it with a vasectomized male, and then it will develop a plug. So if you mate it on Monday, for example, or on Tuesday, for example, 3 days after that, it is going to have a plug, or this is what is the Whitton's phenomenon. And so you want a recipient that is 0.5 days to transfer surgically the 2 cell stage embryos. But you can also culture this embryo into a blastocyst. And for you to do the embryo transfer, then you'd want to have a timed recipient that is 2.5 days after the plug has developed. And you can do this non-surgically, as we do in human beings. So the 2.5 post-plugging is an equivalence of day 5 in human beings, where the embryo transfer is done. And once you have done this, then the hope and the expectation is that you will get some pups. And from this type of service, we have been able to develop many types of, strains of mice, particularly for our experiments in the germ-free status. And that is me doing the embryo transfer. Just to tell you, it is not just theoretical. We are doing it in Bern. But the germ-free mouse would be helpful if you can be able to associate it or to reverse the process that you started with, if you can be able to associate it with microbiota. And this we do by administering the microbes through the mouth, what is called gavage, or per rectum as well, or both ways. And happily, we have known now that you can be able to associate it with microbiota. And you can choose, of course, to take one strain of microbes, which is called monocolonization, or you can have a mix of them. The important thing is that you know which microbe is you are in your mouse, and that is what is called notobiology. Notobiotic means, noto means knowledge, or biology, the normal biology. So you know the microbes that are in your mouse. We have also developed another system, and I'll come back to it, where you associate it with a genetically modified microbe, and then 72 hours later, the microbe is dead, and the mouse is back to germ-free status. I'll come back to that later. Now, I said that you can associate it with either a single strain of microbes, or you can choose a number of microbes. We have developed a model where there are 12 strains of microbiota which have been genotyped, and this we call stable defined moderately diverse mouse microbiota, SDMDM. And this is to try and mimic the actual status of the human being, where we live as associated beings. We are permanently with our microbes. So we want to go back now to that associated being, albeit that we know what we have associated the animal with. And we have seen that this microbiota is stable. We have traced it for 6 years, and it is a stable microbiota, and that gets us close now to making a model of human disease. I mentioned of reversible colonizing microbiota, and in this we took Escherichia coli. This is a publication that we made in 2010. We took Escherichia coli and genetically modified it so that it cannot be able to generate some 2 amino acids, and that is the diaminopimeric acid, that is MDAP, or the D isomer of alanine, and we call it diala. So it cannot in itself, the microbiota cannot be able to generate this, and this one cannot also be provided by the mouse or by the host. And so we provide this externally. So once you withdraw these amino acids, then the microbiota slowly dies out of starvation. And this is what we have done. And you can see here that with the wild-type microbiota, there is stability. That is the black line that you see. And with the blue dots, open circles, you can see that it wanes down, and at around 72 hours, you have a germ-free animal again. The good thing is, this microbiota can be associated with the animals, and this we have found. And we have also seen that the metabolites of this microbiota also penetrate the entire body of the host. And that is what you see, that within 18 hours, that is the graph that you see there, the heat map graph that you see, that all parts of the body, or most of the parts of the body, there is a presence of the metabolites. It is also important to know that much as it is that you have made a good model of either a reversible or a stable microbiota, this is going to be influenced by a number of factors, whether you are checking it at the neonatal stage or during pregnancy or as an adult. During pregnancy, we know that the maternal microbiota play a role to influence the offspring, and this is therefore going to be affected by the maternal nutrition. Maternal nutrition, maternal microbiota will influence the offspring. When you're looking at the neonatal phase, it is the breastfeeding that is going to influence that, plus any other touching that we are doing to the baby during washing or during handling. And at an adult stage, the most significant things are the food, the hygiene, and the antibiotics, the lifestyle factors, so to speak. And depending, therefore, on any of those factors, you can propagate a healthy microbiota or disease, because either of them is going to influence particularly the gut microbiota, but also, of course, the skin and mucosal microbiota. So why do we need reversible microbiota that I've just talked about? It is because we have got a number of questions that one needs to ask. How long do you need microbiota in life for you to effect a mature immune system? And how many of the microbiota do you need? Do you need 1 million? Do you need 2 billion? And also, when in life do you need it? Do you need the microbiota? Is it during birth? Is it before birth? Is it after birth? Is it as an adult? When do you need this microbiota? And with reversible microbiota, you can be able to put it in and then reverse the process at whatever stage of life. What of the monocolonization? When do we need it? We need it because we want to investigate how the association of one microbe with the other influences the immune system, and compare that with germ-free status. This is what I'm trying to show in this thing. And why do you therefore need so many models? It is because we still want to know which of the bacteria that we have so created influences either the immune system, the metabolism, the physiology, or whatever disease model that you have. Is it a consortium of microbiota? Is it one microbiota, or is it an association of one microbiota with the other, or is it just the metabolites that are necessary? And simply to speak, or in summary to speak, this germ-free model that I have just mentioned can help us in answering a number of questions. And this can be right by associating it with the microbiota, or by looking at the metabolites. And we can also look at different types of colonizations to answer different questions in biomedicine. If we were to look at how many institutions have got this, so that we can see the significance, who is using this, this model. You can see in North America, there are quite a number of them. In Canada, there is still one that is not showing. And in Europe, you can see quite a number of them also are using. In Africa, unfortunately, it's just South Africa that had one from 2017. In 2020, they closed down. And so we are saying, and we are arguing, we are availing ourselves to establish one at Kipre. How are we using it? This is a custom-made behavior experiment model. And there's an arena, an open arena here. We have got a T-maze here, and we have put it in an isolator with a camera up there. And we put a mouse inside the arena or the T-maze and see how the mouse is going to behave. And through this, we published this data in July this year. We have now seen that mice that are colonized with this defined flora microbiota have got more stress than those that are germ-free. Other people, of course, have tested different things. This one is where a mouse has got high salt feeding. It is given either saline or another composition of salt, and you give it for 14 days, give it to a jump-free mouse, and you inject it with low dose angiotensin, which increases vasoconstriction, and you can see from there that those that are associated with the microbiota, as we have seen here, will develop, let me see that again. The ones that are associated with microbiota with treatment of salt, they develop hypertension. That is to say, you can be able to have a hypertensive model using a jump-free mouse. Others have done this type of colitis, which is done using the dextran sodium sulfate, and you can see that those ones that are associated with microbiota are rescued from colitis. Those ones that are not have developed full-blown colitis. And we have also got a different model where we are arguing that maternal microbiota, and this is using the reversible microbiota, does educate the immune system of the offspring, and this is what we did. We gavaged the mouse just close to giving birth, and then after birth, the animals were germ-free, and we could see that the immune system of the offsprings were already educated by the microbiota. Others have used the same kind of model where they take twins, one of them who is thin, the other one who is heavier, and you take feces and gavage them to the mice, and you will develop a diabetic model if you gavage the feces from the heavier offspring, and from the thin one, you will develop a thin mouse model. There are, of course, many more things that one can do. I'll rush through. But all this to say that in our facility, we have all these kinds of colonizations. The ASF, this altered Shadler flora, 1.4 means that there are 4 bacteria from the altered Shadler flora. And we have also the SDMDM. There is a group that has now developed the 15 bacteria. That is to increase the number of bacteria just to see whether that one changes. And these are the systems that we have. We house the animals in isolators, as you can see, and any material that goes into the isolators has got to be sterilized using these drums that you see down there, be it the water or the food or the bedding that we shall use, or the nestle, whatever it is, we shall import it using the drum, and sterilization of the sleeve that is interconnecting is by peracetic acid. And you can also do the same kind of experiment using either ISO cages that are on the left or using the IVC cages. I am including this image here because I know Kipre has got these systems in place, and I cannot therefore confidently say it is possible to establish this kind of system at Kipre. At the end of the day, we now know that it is only a mouse that has got wild-type microbiota that mimics or closely mimics the human being's physiology. And so it is back to the colonized mouse or wild-type colonized mouse. And the argument is this, that it is only mice with a wild microbiota that have an immune system that is closer to that of the human being. But I'm trying to say this so that I say it is possible, and this is my last slide, to start from a conventional mouse house, and this is what you have now, and develop an experimental room that has got germ-free animals or associated animals, or a breeding house that has got the same types of animals, either germ-free or colonized. And with this model, you have an opportunity of increasing the value of the mouse in answering the human disease questions, particularly when coupled with the non-human primates that we have here that can also go through the same process. Thank you.
Thank you so much, Dr. Joram. And now we are coming to the last talk. The last talk will be done, I think it's a pre-recorded talk by Dr. Almas Juma, who is a senior research scientist at the Kenya Institute of Primate Research, Department of Reproductive Health and Biology.
Good morning to you all. I am very excited to be presenting part of the research that we conduct here at KIPRE within the Division of Reproductive Health and Biology. My name is Dr. Almas Juma. I am a senior research scientist, and my area of research is within male reproductive health. In line with that, today I will be presenting on the use of preclinical animal models to understand human male fertility. A bit of a biology refresher. The male reproductive system consists of multiple parts, which include the testes, which is the site of sperm production and hormone synthesis. And the testes are housed within the scrotum. We've also got the epididymis, where sperm mature and are stored prior to ejaculation, and the vas deferens, which is the tube that transports sperm from the epididymis to the outside world through the penis, shown here. So there you have your epididymis and testes housed within the scrotum, like I mentioned, and the vas deferens, which is the tube that extends from the epididymis all the way through. And of course, you've got the accessory glands as well, shown here in green, the seminal vesicles and the prostate gland, which contribute multiple secretions into what we call semen that carry the sperm. So overall, reproduction in both males and females involves the hypothalamo-pituitary gonadal, or HPG axis, involving a top-down regulation and feedback mechanisms that ultimately control gamete production and hormone synthesis by the gonads, which are the testes and the ovaries. So you've got up top the hypothalamus, which regulates the pituitary gland, which then regulates the gonads via production of hormones such as luteinizing hormone and the follicular stimulating hormone. And ultimately you get production of the eggs and the sperm, and hormones, which also are responsible for feeding back to the top regulators to hence regulate the HPG axis. So in a nutshell, male fertility involves the formation of healthy sperm in the testes that are capable of successfully fertilizing an egg, followed by formation of a healthy embryo, post-fertilization, and a healthy baby in the end. Now, male fertility is an extremely taboo topic, and in our society, much so, as well as the globe at large. The data is sketchy and most likely an underestimate. However, infertility affects approximately 15% of couples worldwide, which translates to about 1 in 7 couples. The accepted definition of infertility is the inability to conceive after 12 months of regular and protected sex. And an important fact that most are unaware of is that males are actually responsible for up to 50% of infertility cases. And to exacerbate the matter, 20% of these cases are idiopathic, meaning that the male is infertile with abnormal semen parameters, but everything else, such as the hormones, there's no obstruction in the vas deferens, everything else appears normal, and yet the hormones are normal, and yet the individual has abnormal semen parameters, meaning the cause of the abnormality, the cause of the semen abnormality is unknown. Another 10% of cases are unexplained, whereby the man has normal semen parameters, the female partner also has normal fertility parameters, but the couple remains infertile. In order to give you a snapshot of male infertility within our society, we retrospectively analyzed data from 115 men that visited an infertility clinic in Nairobi alongside their partners. The men ranged from 21 to 56 years of age, and only 54% of them, 54% of the men, of the 115 men, had normal sperm parameters, also known as normal zoospermia, while the rest, so for 46%, almost 50%, almost half the men, had a range of different sperm or semen abnormalities listed, shown here in the graph. This data is important in highlighting that relatively young men in their 20s are suffering from infertility as you can see indicated here in this pie chart. So the abnormalities here are grouped singularly. I won't go into too much detail of the definitions of them, but they mostly range from abnormal sperm count to abnormal morphology, etc. And now, without going into too much detail, this is just to show you that a particular individual could have either a singular abnormality or a combination whereby they could have up to 3 semen abnormalities. So, for example, an individual could have defects in sperm motility, morphology, and or low sperm count, so all 3 combined, as is the case shown here with the red arrow. These 11 individuals have what is called astenoteratospermia, whereby they have abnormal morphology, low sperm count, or absent sperm, and abnormal motility as well. So as you can see from this data from just a single clinic in Nairobi, this highlights that male infertility is a very real and relevant issue, and this warrants more attention, more advocacy, more medical resources, and of course more research, which in our case here at KIPRE is preclinical and animal research to inform human male fertility. At KIPRE, we conduct preclinical, biomedical, and translational research using animal models. We are guided by local and international standards, including OLAO and ALAC. And our models include the non-human primates, as well as mice and rabbits. And I can't leave out, of course, the snakes that we have here at KIPRE that are used for antivenom research, not reproductive biology research, but of course I have to mention that we have them as well here as animal models at KIPRE. Now, mice are commonly used to investigate the role of genes in male fertility. This particular project that I've given a snapshot that looks very thorough here. Without going again into too much detail, this particular project investigated the role of pleomorphic adenoma gene 1, which is a transcription factor, which I will refer to hence going forward as PLUG1. So PLUG1, we knocked out the PLUG1 gene in the mice and replaced it with a reporter gene known as LACZ or beta-galactosidase. This enabled us to genotype the mice, as shown here on the gels. And the yellow arrow, for example, depicts a heterozygous mouse, which has both the PLUG1 gene detected and the LACZ gene. And when you do a multiplex PCR, you're able to clearly see the 2 bands belonging to the 2 different genes, indicating a heterozygous phenotype. And shown in red here, it's showing an absent PLUG1. Here, the gel is clear, and a present LACZ, indicating a complete knockout of the PLUG1. So this particular mouse is a complete knockout. So breeding studies, if I can refer you to these diagrams at the bottom here, breeding studies have shown that mice have reduced fertility. Rather, the knockout mice have reduced fertility. The knockout males have smaller testes, as you can see here. That's the wild type and the knockout, and they're smaller. The testes and the epididymis shown here, this long tubular structure. Both the testes and the epididymis are smaller in the knockouts compared to the wild type relative to body size. And in fact, the epididymis is malformed. You can see the wild type structure down here, if I can show you here at the bottom right. The wild type structure is complete. However, the knockouts have a bit of a malformation, indicating a role that this gene is required for proper formation of the epididymis. Additionally, these knockouts have reduced motility, and more specifically, they have reduced progressive motility, which is crucial in terms of sperm function and the ability to get up, to fertilize an egg, to swim and go fertilize an egg. So the human and mouse PLUG1 proteins have 96% similarity, inferring similarity in function. We performed RNA sequencing to compare the transcriptomes of the knockout and wild types and heads, and found several upregulated and downregulated genes in the knockout. And some of these downregulated genes in the knockout testes are genes that are involved in spermatogenesis and testosterone synthesis, showing that the presence of this gene PLUG1 is involved, is required, or rather PLUG1 in a wild type is required in processes that regulate sperm and testosterone production. Very briefly, the presence of the LACZ reporter gene enables us to localize the in situ expression sites of PLAG1 across the HPG axis, so within the hypothalamus, within the pituitary gland, and within the testes. So these little blue dots that you're seeing are all representing everywhere that the LACZ gene is expressed, and the LACZ gene has been placed in place of where the original PLAG1 gene is, hence enabling us to identify cell types that expression sites of PLAG1, which we then combined with immunohistochemistry for specific antibodies for specific cell types to identify specific cells that express that gene PLAG1. So mice, overall, with all that data, it's just to emphasize that mice are versatile and they can be successfully used in gene manipulation studies that provide what I like to call a goldmine of data to further elucidate gene functions in crucial physiological processes. The results of this study have been published in several journals shown here for your reference. I'll now pivot to our much larger animal model, the olive baboon, known as the Papilio anubis. Due to the size and anatomical similarity to humans, baboons serve as an excellent model for reproductive studies. So I'd like to highlight one special characteristic shown here, which is the sex skin of the female baboon. This is a prominent and externally visible feature which allows non-invasive staging of the menstrual or estrous cycle, because the characteristics, as you can see, change according to the cycle, the estrous cycle, or the stage of the estrous cycle. For example, the swelling, the very prominent swelling you see here indicates ovulation, and this special feature enables ease in breeding and other fertility or reproduction studies using this model. So Dr. Tunganyacheo, shown here, worked with a team from Belgium to develop a baboon IVF model, working mostly with the female baboons and conducting various procedures, including ovarian stimulation, oocyte retrieval, menstrual cycle synchronization of the baboon in the baboon model. But of course, this is all focusing on the female, but IVF requires the male gamete too. So Dr. Atunga, and alongside Dr. Madera and Dr. Chai, also established protocols for baboon spermatology, which they successfully published as shown in the snapshots of the articles on this slide. Now, unlike the smaller animal models such as mice, which often require sacrificing the animals to obtain the sperm for seminology, baboon sperm are easily acquired by electroejaculation. So pictured here, a normal sperm, an abnormal sperm from one of the Kipper studies. And you can see the normal sperm here on the top left again, and an abnormal one here with a coiled tail, one with an elongated head here, which is abnormal, and one with a bent neck here. So these malformations, of course, prevent normal function of the sperm, and this was nicely shown using the baboon model by our researchers. So using this established and published techniques, they were able to study effects of Catha edilis, also known as cat or mira, shown here, which is a recreational drug commonly consumed in East Africa and Arab countries. And caffeine, shown here, was also investigated, or the effects of caffeine on sperm parameters. And please take note of these studies, of these papers shown here, if you're curious about the effects of these compounds on male fertility. But overall, the MIRA study showed that cat or MIRA has negative effects on semen parameters and reproductive hormones, including testosterone and prolactin, hence contributing to male infertility. Conversely, they showed that caffeine enhances sperm motility and may be beneficial in IVF studies or protocols in the baboon model. So the overall takeaway messages are that from this presentation are that animal models are valuable for male fertility research, ranging from basic all the way to preclinical studies. I would also like to mention the huge funding gap that unfortunately exists when it comes to reproductive health research, especially male fertility. And I would like to urge for more scientists to take on this important area of research that is directly impacting our society, as I indicated from our retrospective analysis of the human data. So with that, I would like to acknowledge the entire KIPP Reproductive Health and Biology team pictured here, as well as to thank the entire institute, the Ministry of Health, and my supervisors, Dr. Jael Obiero, Dr. Bert de Groof, Sylvia Groman, and Professor Moira O'Brien. If you have any questions for me or would like to get in touch, my contacts are included on the slide. Thank you so much for your time.
I'd like to thank everyone for being very patient and keeping up to the end of the session. And as I told you, there's a cocktail after this. But before that, I'll call Dr. Atunga to give the closing remarks.
I think the last time I was here, it was morning. So now I can say good evening or good night. Is it night or evening? So I want to take this opportunity to welcome, not really to welcome, but to thank everyone for being patient today. It has been a long day. And when I look at the people's faces, I think they were not like in the morning, because I saw you in the morning, and now I'm again seeing you in the evening. So, but overall, I think the day has been very successful, and we have had quite a lot of engagements from policymakers to experienced and distinguished speakers, and I think we have learned a lot. And from today going forward, we are no longer strangers. You know, in the morning, everybody was new to each other, the people around them. But I think we have listened, we have talked, we have interacted. And now, as we move forward, I think it will be easier to be able to interact and discuss more. So, I know the program is quite heavy, and probably we have tried to meet your expectation, and maybe not really fully. There are people who came here, they want to listen only immunology, only parasitology. Others have come to listen to snake bite. They have heard about antivenom. And then others have come to listen about AMR. And maybe they look at the presentation today and they're saying, all these presentations, what we're expecting, we haven't seen it. But I want to promise you that if you look at the program from yesterday, from tomorrow, you'll be able to see that there are parallel sessions, and you'll have the freedom to really join where your passion is, and you'll contribute quite effectively. Secondly, I also want to apologize for the delay. We had a bit of delay in the morning. Is because we were also waiting for the PS to open the session for us. So again, you were patient that morning, but we tried our best to catch up. But even so, you know, just apologizing only is not enough, isn't it? Just being apologetic, we are apologizing is not enough. Sometimes you need to do something extra, more than apology. And there is a cocktail. I think a cocktail is going to clear all the things, all the delays and everything that happened this evening. So, and the cocktail I want to promise you is a very good cocktail that you will enjoy, and then you'll have calm sleep, and then tomorrow when you come, the issues of delay and apologies will not be there. I hope so, isn't it? Yes, yes. So it will be very good. So we have cocktail. The cocktail is on the 10th floor. You know, the 10th floor is a very good place where you will be viewing. I don't know whether the animals will come close at night, whether we are able to see them. And not only animals, we can see other things. So there is a very nice view for that purpose. We were not able to have some question and answers. I think normally it's very important we have those sessions. And the way the program was designed was that we have 15 minutes presentations and 5 minutes question and answers. But I think maybe from our side, it wasn't very explicit. And I think all the presentations that came, they already have had like the whole of the 20 minutes. So really we tried as much as possible to save time for question and answers, but it was a bit difficult. However, I will encourage that during the cocktail, now that we have seen the speakers, if you had a specific question, maybe you can also use that time to ask questions. I know the online presenters will not be there, but at least for those of us who are around, we can have some interaction, some questions. And I already saw that happening during coffee time, where I had one saying, I wanted to ask you a question on this, and they had some conversation, and I think is going to be very, very good. So tomorrow, as we prepare our presentations, kindly, the presentation is 15 minutes and 5 minutes. Let us reserve 5 minutes for question, 1 or 2 questions, or even some comment. We have some companies with us. Today we have Change Biotech. I don't know whether anybody visited the 2 companies that are here. I know the time was very tight. Most of the time we just went for coffee and came back. But on this side we have some 2 companies, and maybe others will come. And tomorrow we shall have more people actually here who are attending the sessions. So we have 2 companies, Change Biotech and F&S Scientific. So I will encourage, as we get out, going to the cocktail, at least visit them, have a chat with them. They have some new technologies that we may require for our work. So I don't want to say so much this evening, especially when people want to go for cocktail. I just want to, again, appreciate you, and then say that please look at your neighbor. Look at your neighbor. Nobody's looking at the neighbor. Look at the neighbor. And say, ask the neighbor, are you so exhausted? So I just want us to appreciate ourselves for having stayed for a long time. And we shall do one club, which is called Factorio Club. We shall do 5 Factorio. You know, 5 Factorio means you clap 5 times, then 4 times, then 3 times, then 2 times, then 1 time. And then once we finish with the 1 time, we shall have closed for today, and then we go for the cocktail. Okay? So shall we rise up? So 5 Factorio, isn't it? So we shall go 1, 2, 3, 4, 5, 1, 2, 3, 4, 1, 2, 3, 1, 2, and then 1, and then the meeting will be closed. Then we go to 10th floor for the cocktail, right? Okay, so let's go. We start. Okay, first of all, prepare. We have to prepare ourselves. Preparation is this way, yeah? You need to warm the hands a bit. Yeah. Okay, then we go. Thank you very much. The day is closed. Now we go for cocktail.